You ready?
Okay, hi everyone. Thank you so much for being here. We're going to do a quick intro and then move into the first part of our conference. So welcome to the Harvard OpenBio Future of Biology Conference, co-hosted with the MIT BioMakers. We're so thrilled to be hosting you all at our first in-person iteration of this conference. My name is Audrey. I'm the Vice President of OpenBio, and I'm one of the directors of this conference.
Hi, my name is Ayush. I'm co-president of the Harvard Open Bio Lab, and I'm also a conference director.
And I am Hugh Hankinson. I am the other co-president of Harvard OpenBio.
So 3 years ago, we launched the Harvard Open Bio Lab with a pretty simple mission: to empower student biologists and build a student community at Harvard that uses biology to move our world forward. So we at the Harvard Undergraduate Open Bio Lab, we support student-led biology research by providing resources, funding, lab space, mentorship, and advising to help undergrads turn their research vision into reality.
We seek to make exciting and meaningful biology research accessible to all, and our team is committed to increasing access to research opportunities for underrepresented groups in science. We back projects that have the potential to make transformative impact on the world using biology.
Yes, definitely that. And to fulfill this vision, we're super excited to be hosting our second annual Future of Biology conference along with an amazing team from MIT. Shout out to all of you that are here upstairs. Our vision for this conference has been to break down barriers to biology by democratizing scientific experiences and connecting bright minds like you with each other in order to foster the future of biology.
Before we get started, we have some thank yous to make. We'd like to thank our entire team at the Harvard Open Bio Lab, our engagement team. They've been the backbone of this conference. Our lab team, they're going to run an incredible wet lab workshop today. Highly encourage you to attend. And our finance team for helping us achieve the support needed to put this incredible event on. We also want to thank the Harvard AV and Events team for running all the AV that you'll see here today. And we want to thank our sponsors, including the 1517 Fund and the Harvard MCB Department.
And lastly, we want to thank you all so much for coming today, those of you in person here in Cambridge and those of you on Zoom across the world. And now it is my pleasure to introduce Sean Meng, who will be introducing our first keynote speaker, Dr. George Church.
Hi, everyone. My name is Sean. I'm a second year at Harvard studying neurobio, and I serve as co-director of engagement here at Harvard Open Bio. You guys may have been getting a few emails from me. I apologize for the spam. But thank you all for being here. We've been working hard toward this day for the past couple of months. And to kick things off, I have the pleasure of introducing one of science's heroes, Dr. George Church. I was told that, Dr. Church here needs no introduction. But just very quickly, George leads synthetic biology at the Wyss Institute, where he oversees the directed evolution of molecules, polymers, and whole genomes to create new tools with application in regenerative medicine and bioproduction of chemicals. Among his recent work at the Wyss is development of a technology for synthesizing whole genes and engineering whole genomes far faster, more accurate, and less costly than current methods. George is a professor of genetics at Harvard Medical School and professor of health sciences and technology at Harvard and the Massachusetts Institute of Technology. He's director of the U.S. Department of Energy Technology Center and director of the National Institute of Health Center for Excellence in Genomic Science. Join me in welcoming Dr. Church. Thank you for being here with us, George.
Oh, thank you very much. I am going to, it's really terrific to be here. You've got some of my favorite topics. I'm going to share my slides in just a second, and also hope that there's time at the end, and sufficient enthusiasm for some Q&A, because I think that's my favorite part. So tell me if you can see me and my slides at this point.
Yes, we can see both.
Yeah. Okay, and should be presenting one slide now, and I have a lot of thank yous for the various people. along the way here, and institutions like the ones here that helped get these ideas out into out of the lab and into the world. At the bottom here is my potential financial conflict of interest list. It's a website because it doesn't really fit in the slide or two. And so you may wonder what this spinning thing is. This is transfer RNA, which I've been working on now for 5 decades. When I used to look at it 5 decades ago, it looked like the thing on the left, which is this black and white static not 3D object. And this is what roughly what I looked at that time, 73. And then 10 years later at the Harvard Biolabs, there I'm working there with 4 amperes and 5 kilovolts, not at the same time. I teach a course that's, I think, resonating with this group here today. It's called How to Grow Almost Anything. It's taught at both in person at Harvard and MIT. But also worldwide to about 500 people in all these locations here on the map, including distribution of up to 32 robots, thanks to Opentrons and thanks to Nuclera. They can not only talk to each other, but they can do experiments in each other's labs remotely. So that's that course was actually written up in Nature Biotechnology, which is the first time one of my courses has been written up in a major journal. Now we're getting very close to DNA Day, which is an annual event now for decades that my group has celebrated in various ways, including a whole series of what we call GET conferences, genomes, environments, and traits. So technically the day is 25th of April, resonating with the publication of the double helix by Watson and Crick and Rosalind Franklin and so forth, and also with various announcements on the Genome Project. And this was a unique event in 2010 where we got together everybody who had been sequenced, almost everybody that had been sequenced and had their own sequence and had announced that they have their sequence. And it was just this small group here. And it included people like Harvard professor Skip Gates and Jim Watson and a lot of the CEOs of the companies that enabled this sequencing, and the PGB10. This was open. So the 2 themes today is openness and the future. And this is the past, but this is how we made personal genomes open, including their medical records and their cell lines. And that's still a very useful resource, as you'll see. Almost every experiment that I'll show you having to do with human cells were done with Personal Genome Project cells because they were properly consented and available for commercial use. Another so that's one open resource is personalgenomes.org. Another one is pged.org, which is for education of Congress and screenwriters and so forth. A third one is adgene.org, where we have we've distributed both CRISPR and transcription factor collections, and this is these are shipped at cost, essentially the cost of storing and maintaining and shipping by Adgene, this nonprofit, and the transcription factor library is somewhere between 1,000 or more, and within just a few months of us depositing and publishing our first CRISPR paper, and the same thing goes for Feng Zhang, one of my ex-postdocs who independently has done terrific work on CRISPR. Anyway, both of us deposited. A couple of months later, there were 10,000 users worldwide. So that's an example of openness. We had 2 early wild ideas for sequencing. I'm going to just go through them quickly because it's about democratization of sequencing. It was when we started talking about sequencing humans genomes, it was $3 billion, and I felt it was completely unacceptable, even as a goal, especially as a goal. And so one was nanopore sequencing, which is kind of this weird hybrid of patch clamp for neurons and Coulter counters for particles, plus DNA sequencing. And now there's 3 different versions of this, at least. I think we're the only group that's participated in all 3 of them. Oxford Nanopore, Genia, and Roche, and Electronic Biosciences. And so this has been democratized to the point where it not only can small enough that it can fit 4 of them easily in the International Space Station. You can see it in the lower right of Kate Rubin's photo, but it also goes out into the Ecuadorian rainforest and field studies all over the place. Here you can see it next to a pit viper. So this is very inexpensive and small size. The other idea, in addition to nanopore sequencing that we had in the 80s, was next-gen sequencing using fluorescent microscopy. A little video that's going on there is a single fibroblast cell from Personal Genome Project, remember? And this was, every one of those dots was a single RNA molecule, which gave various colors, ACGT, 4 colors, and each cycle of sequencing gives it, and you could play it back computationally. So each little bar there of colors is a barcode that tells you which RNA you're looking at. And so this has gone from just plain microscopy to next-gen sequencing, and then from there to spatial omics, which means you can actually get the 3D structure of tissues and brains and so forth. And there are 2 major ones that we've worked with, which is 10x Genomics and Bruker Vitara. And we just had a preprint out there with this PMID from Johannes Stein, where we're getting 3 nanometer precision in these sections. This started as a Nikon microscope. It was open source. It opened hardware, wetware, and software. And the combination of these efforts at democratization has brought the price down tremendously, even faster than Moore's Law for computers. This is about factor 20 million over a fairly short period of time, maybe 10 or 15 years, where we have gone from $3 billion for kind of a crummy genome, if I could use technical word there, a haploid, meaning only 1 genome rather than the 2 that you get from your mother and your father. Fast forward to today, and now it's in the lower right here. It's been $249 now for 3 or 4 years, rather than $3 billion, and that's for a diploid genome, a high quality genome. But that's great. We brought down the cost of reading and writing DNA about 20 million fold. What about therapeutics? Abbreviated here, TX. Isn't that going up? And indeed, it's very high for certain categories of drugs. So in the upper left, you see the gene therapies are the most expensive category of drugs, now getting into the $3 to $4 million per dose range. This is because the expensive R&D and clinical trials is amortized, spread out over a very small denominator of, hundreds of people per year for some of the rarest diseases, like MLD. But there is hope in a slightly adjacent field, which is pandemic vaccines, where the major top 5 vaccines were formulated as gene therapies and manufactured in that way, and either viral capsid with double-stranded DNA or lipid nanoparticle with single-stranded messenger RNA. And I think most of us have taken that. In fact, 5.6 billion people have taken this. And the result was, instead of $4 million, on the order of $20 per dose, maybe even as little as $2 in some cases, and it could go down in the future. So this is the economy of scale and the urgency of the situation that brought it out in less than a year through full approval. Now, another thing that we can do is we can make organisms resistant to all viruses. So that was a particular case where we could make a specific vaccine to a specific strain of a specific coronavirus. But what if we can make, and you know that we get a new strain of influenza every year. So what if we could make something that is resistant to all viruses, all naturally occurring viruses, no matter what? And that was one of the goals that we had for genome recoding, where we changed the triplet codons. So there's 64 triplet codons, A, C, G, and U, various combinations. They encode the 20 amino acids plus stop codons. And we wanted to change those so we could do biocontainment, number one. Number two, non-standard amino acids. And number three, this multivirus resistance effect. And thanks to Harris, Farron, Mark, Dan, and Nilly, shown here at the bottom right, and the papers that they wrote. And at least one company has come out of this, Grow Biosciences, thanks to Dan Mendel. This is the codon table. Some of you may be familiar with this. You've got three, you got first base, second base, and the third base, A, C, G, and U. And then all the numbers are the number of times that codon, that triplet, appears in protein coding regions in the E. coli genome. And we changed first the red one, 321 changes genome-wide. And that got us most of what we wanted in that previous slide, the biocontainment, the virus resistance, and the non-standard amino acids. And then Jason Chin's lab did two more in green, and we're just now in the process of finishing up 6 more codons, making a total of 7, including the stop codon. And here was what we had been hoping and imagining on paper, but now it's reality, was that you could get resistance to all viruses, all natural viruses that we've tested at least, which is almost all the lab strains and a bunch of new strains that we got from environmental isolates like sewage and farm waste. This is thank you to Akko, Svena, Reagan, and others. Here, what we did is we swapped 2 of the 6 serine codons into leucine amino acids. So instead of coding serine, they code leucine. And let me just assure you that serine and leucine are about as far apart as they can be chemically. And so when you swap those, it messes up multiple positions in every gene of every virus that we've tested. And so this works like a charm. You can see out on the lower right that you can get strains that are resistant to very high input virus, no output virus. For biocontainment, we can put in new non-standard amino acids to get biocontainment. Here's an example of one of those where we've solved the crystal structure thanks to Barry Stoddard, and it is what we predicted it would be. And the next step up, we haven't gotten there yet, but we got some preliminary data I'll show you in just a second, is to extend the translation to not 64 triplet codons, but 128 triplet codons. By having 2 systems, one of which is mirror life on the right and normal translation on the left, and they're orthogonal by keying into the 3' end of the tRNA. I'm getting into the weeds for the cognoscenti in the crowd. Normally they end with CCA, 3' amino acid. In our case, we can do CGC, CGA, and then the amino acid. So this is some preliminary data from that project. We're obviously not anywhere close to done, but we have 12 out of 20 of the tRNAs are accepting amino acids at this point to the CGA. So these are the only tRNAs we know of in the world. Every organism has tRNAs, but they all have CCA at their end. Now they have CGA as well. Now, when we talk about therapies, we have some choices, which I think they all basically can be thought of as gene therapies, but genes can make epigenetic changes, or genes can encode proteins, or genes can be the key point of cells. You do the gene therapy outside the body and then put it in as a cell or as an organ. So I'm going to just give you some flavor for that. Here are 3 of the groups that have benefited from the epigenetics. So this is the epigenetic slide. We sometimes call it the TFO for all the transcription factors, which are DNA binding regulatory proteins. We not only know what the transcriptome is, what the RNAs look like in multiple target cell types differentiated from stem cells. We can not only read those, but we can write them. We can go to any of these, and to our pleasant surprise, the typical recipe is 4 days with close to 98% conversion from the stem cell into your favorite cell. And I'll show you some examples. Here's some where we did endothelial cells, neurons, and fibroblasts. Each of the vertical lines here is a different transcription factor recipe, showing there's multiple ways to get each of these categories of cells. They'll seem to get attracted into a common developmental area. And this is my thank you here to Alex and Paris, who not only wrote this Nature Biotech paper, but also co-founded GC Therapeutics. Now here's an application where we took, and you'll see in a moment another one, but this is where we took made in vitro ovarian follicle-like structures, where the key player here is granulosa-like cells, which support oocyte development. So in an in vitro fertilization clinic, the mothers are given doses of hormones like gonadotropin, and this is quite painful for many, but not all women. And so if you lower the dose, much less pain, and byproduct is it's 3 times cheaper as well. And so we've done that, but the problem is the oocytes are less mature, so they have to be mature outside the body, which we use our stem cell derived granulosa cells. And this works. This looks very promising in the clinics. So this Christian Cram was a graduate student in my lab, along with Mirik Smila, founded Gameto, and is now there full time. Orchid Health does whole genome sequencing of all the IVF embryos to make sure that none of them are, or to pick the ones that are least likely to have not have spontaneous abortion and other serious postnatal diseases. Way postnatal, typically, there's chronic kidney disease, and this impacts a surprising fraction of the world. Sometimes it's covered up by other diseases, but it's at the root. It's a horrible risk factor. So 10 to 14% of us have it. 800,000 in the United States have end-stage kidney disease, but only 20,000 per year get transplants. Dialysis is also a painful experience, 4 hours a day, 3 days a week. So how do we increase the number of donors? So we have a donor shortage, 25,000 per year when it should be 800,000. We have the first transplant occurred at a Harvard-affiliated hospital in 1954 with an identical twin. Unfortunately, there aren't that many identical twins to act as donors. There are, to each other, there is immune suppressants, and essentially all transplants now are done with immune suppressants. Well, how can we increase the quantity of organs? So these are some of the pioneers. I'm not going to go through all of them. The 3 on the far left are significant because they're the ones that invited myself and Luhan Yang, I'll mention in a moment, to apply our new CRISPR technology to this problem, where clearly needed multiple gene edits, and I'll show you just a moment. This dates back to 1963 with chimpanzees, which didn't work that well then, and it's off the table for ethical reasons anyway. So we can now, so this talk is about openness and future, and the future of editing genomes is multiplex editing, or even writing genomes, as I've shown you. Right, we wrote genomes to get virus resistance. And so we can think of the organs, we want the organs not only to be readily available and inexpensive, we want them to be enhanced in certain ways at a minimum so that they don't fail for the same reason the original organ failed. For example, whatever the risk factors were, for example, pathogens. And so we thought of 7 different, they could be considered enhancements in the same sense that vaccines are enhancement relative to our ancestors who didn't have them, who are now resistant to many, many major pathogens. Not all, but they're examples of every kind of enhancement there is demonstrated in animals, including extreme resistance to cold, meaning it's minus 55 degrees, and to dryness, meaning complete desiccation down to 3% water. They can be resistant to a variety of sources of DNA damage, to immunity. That's the key one for rejection. They can be pathogen resistant. And of course, you don't want your transplanted organs to senesce prematurely or get cancer if you have any control, which you do when you're transplanting from an animal. You can't change the germline. We're not permitted to change the germline of humans, and ill-advised in any case, but with animals you can. And so here's an example where you can get human tissue in an organ-like structure, organoid. It's resistant to the original autoimmunity of multiple sclerosis-like disease, and this makes normal myelin. This is it's circling the axon, just like you have in your white matter of your brain or your spinal column, and this will restore proper function by this functional assay in a mouse that had, or mice plural, that had a autoimmune demyelinating disease. Again, thanks to Alex and Peristu. But for the pigs, we wanted to resist immune rejection, and this required 69 edits altogether. Now, most of those had to do with pathogens that were contained within the pig genome of all pigs. We did 59 edits to get rid of the endogenous retroviruses, but to resist immune rejection, 3 top killers were the sugars that were present on pigs and not human. And we put in 7 human genes so that, because the pig genes were not compatible with our blood complement and blood clotting and innate immunity and ischemia and so forth. And so that combination has now been put into patients for a liver experiment, and now there's one patient, first in the world, that has a kidney that seems to be functioning fine now for a month, and he's doing things he hasn't been able to do for years. A very happy camper. This was started thanks to Luhan Yang here in the photo on the lower right. She was a graduate student and postdoc in my lab, and she and I co-founded Chihan Biotech in Hangzhou and Egenesis here in Cambridge, Massachusetts. And there's a series of papers that she and Yu and Chen all contributed. Now, so that's immune rejection and some pathogens, the endogenous retroviruses. A broader set of pathogens can be avoided because they're human-specific pathogens, for the most famous ones being smallpox, which we've eliminated, and polio, which we're getting close to, and then all these other ones. Okay, that's great. So we've got endogenous retroviruses, we've got human-specific pathogens, but we'll get to in a moment resistance to all viruses. I already showed you this in the context of an industrial microorganism. We're trying to do exactly the same thing again, but now in pigs, to make them resistant to all viruses. This is a genome-wide thing, which, as you can imagine, is quite a bit harder in pigs than it was to do an industrial microorganism, E. coli. But it should work the same way. And finally, we have aging and cancer, cancer being a typically age-related disease, although there are occasionally early onsets still age-related. And these are some of the discussion points for aging. These are not pathways. They correspond to pathways we know quite a bit about in biochemically. But the things that you've probably heard of, things like caloric restriction and telomeres and epigenetics and mitochondrial dysfunction and so forth. And we have to address all of these at once to really have an impact. Because if you just do one of them, like say cancer, it's been estimated that's an average life expectancy change of 2 years, which is better than nothing, but it's not the economic and social and medical wellness that we're looking for. We tried out 45 different gene therapies and did every possible combination of the 3 best, the ones that were also spread through the blood, which made it very easy to do a delivery, gene therapy delivery to the liver, followed by blood delivery of the proteins, getting every cell in the body, basically. Noah Davidson published this paper in PNAS where he had shown here are 3, but we had 4, soon 5, now 7 to 9 different age-related disease models, either accelerated aging or actually using old mice, as you'll see in just a moment. Noah was a postdoc in my lab. He was co-author on 3 of these 4 papers, all of them using AAV as a delivery vehicle. And we really wanted to use diseases as the metric rather than biomarkers, which you could get right but still die of old age related diseases. And also not only we wanted to avoid biomarkers, we also wanted to avoid longevity because this requires very long clinical trials for humans. But nevertheless, in the animal trials, we let them, after we're done with the reversing age-related disease, we let them live to see if there is a longevity effect, even though that's not what we're going to the FDA with. And sure enough, there is for one of our combination therapies, not the one I mentioned having to do with liver, but this one is ideally targeted at most of the nucleated cells in the body. Here, this is still very primitive delivery, but we've got with a cohort of mice that are basically at their midpoint of longevity, meaning that where half of the mice are dead. If we dose at that very late point in their life, we still get a very significant improvement in their outcomes, where we get a shift, the blue line being shifted about 50% to the right towards longer survival times. So percent survival versus number of weeks of life. So that's the Rejuvenate Bio story, and so they hopefully will have at least 2 triple gene therapies in clinical trials very soon. I just want to end on this notion of machine learning. I'm betting that some of you in the audience are heavily involved in this in one way or another. Here's, I think, a great combination, which is having these big libraries. Originally we called them mega libraries. Now they're trillions or tera libraries. Combined with machine learning. So it's kind of like a natural computer together with a silicon von Neumann computer. And we showed, we being Pierce and Eric and their colleagues at Dyno Therapeutics, that you can target the brain about 100 times better than the previous best. So the green brain at the upper right versus the magenta previous best, 100 times better. And we avoided the liver, which is about a 10-fold effect. And you can do this by loops of synthesis, selection, where each virus apportions itself to different body parts, different cell types, and you can take samples of each of those from non-human primates, 3 different species we used, and then loop back into machine learning and keep going, eventually doing effectively trillions of designs. And this is true not just for Dyno Therapeutics, but for 5 other, maybe 6 or 7 at this point, startups from my group that are using this combination of these gigantic libraries that you can make 10 trillion design genes plus machine learning to get nucleic acids and proteins that are optimal for whatever your goals are. And we use junk DNA. So some of you may have heard of junk DNA. This is these are some papers where it's associated with senescence, but there's all kinds of things that has past influence on, and I'm going to talk about the future influence of junk DNA. We can use we can put signals in the junk DNA that allows it to rearrange, completely scrambling the genome. We've got over 7,000 different rearrangements we've obtained in 2 different kinds of human cells, haploid and diploid. It's just really amazing what you can do at the whole chromosomes, moving between chromosomes and so forth. We can also store data in this junk DNA, DNA that doesn't have any, where you can put information there without disrupting physiology. We can measure, monitor physiology, and record that physiological effects via CRISPR or a variety of other methods. We can put it into the junk DNA or DNA that's not hurt. And this goes back to 2002 when Jay Shanduri was graduate student, MD, PhD, and postdoc in my lab. Now he's been full professor and director of an institute in Seattle, University of Washington. We initially encoded my book into DNA, but now we're encoding the physiology of organisms. And we estimate that with our first methods, we got up to a fraction of a terabyte, trillion bytes, and we could get probably beyond 1 exabyte. That's more than 10 to the 18th bytes of information in just a tiny fraction of a tiny organism. The mouse, which weighs 30 grams, we can get in a billionth of that. We can fit in, we already have fit in 0.2 terabytes. And we can tell things about how the brain develops and so forth. And then just very speculatively, if we can measure physiology and record it in DNA, in principle, we can make very inexpensive cameras that because your eyes and ears and everything are physiological, and so what they're recording could be encoded into DNA. So this is very speculative, possible future. That's the theme of this. And then the final topic is how wolves save beavers in Yellowstone. They scared away the herbivores from the willows. The willows got bigger, they attracted the beavers. They started knocking them down, making dams as they do, and that encouraged all of aqua culture. We have a similar thing, we think, where the keystone species, like a wolf, could be important in the Arctic. I won't go through the full argument, but Asian elephants like snow. You can see there with a big snowball, and we have a problem in the Arctic. Last time I was in Siberia was the first time it didn't refreeze after the summer thaw, and here's my friend Sergei Zimov demonstrating how much methane there is in all the lakes and the ground water as well. Methane is 80 times worse than carbon dioxide, and there's 1,400 gigatons compared to the mere 10 gigatons of human activity. So we're looking into ways that we can help conserve endangered species and restore environments. So this is another open strategy. This is open technology for conservation. Colossal will make money some other way, but this is more or less a gift to all these different partners that are working with us at Colossal on endangered species. I will open it up for questions there. We have time.
Yeah, let's do that. Thank you, George, for your remarks. Raise your hand if you have a question. I'll run the microphone to you, and then we can get started with that.
Thank you. That's a very informative talk. And I know that genome editing, as you said, is the future of a lot of clinical application. So I was also wondering, what would be your thoughts on the potential strategies to control for off-targets, editing, and related toxicity related to this treatment?
Well, that's an interesting question. Actually, most of the things I described for gene therapy are not CRISPR or editing formally. I mean, they're usually inserting a new gene. Nevertheless, you can have off-target insertions. In fact, some of the insertion isn't even targeted. So far, there are very few examples of it, at least after we cleaned up our act in the year 2000. So it's been about 20 years. The off-target is extremely rare. When you go through germline, like we do with the pigs, is one way of delivering new combination of genes. There you have the luxury of being able to analyze the genome before you use it in a human being, and you can sequence it and show that there are indeed zero off targets, and everything on target that you wanted is there. So, and we've done that. We've sequenced many, many pigs along the way. And you can do the same thing for human stem cell therapies that I illustrated. So I'm not saying it's not a problem. I think it's a nice example where we raised the alarm early on before for CRISPR at least, and we'll keep looking at it. Thanks for the question.
There.
Hi, Dr. Church. I'm just wondering, for editing with CRISPR-directed enzymes, but without a double-stranded break, such as using the dCas9 to direct a uracil N-glycosylase to direct a U to C mutation, would you think we would be able to expand the sets of nitrogen bases that we can edit with that strategy, with that type of strategy?
Anything's possible. Just point out that it's not the uracil glycosidase that's the main delivery, it's the deaminase. And there are both A and C deaminases that have been achieved with great effort from David Liu's lab. But I think the direction the field is going is slightly different. I mean, although the lesson is let many flowers bloom, let a lot of different methods. But the powerful thing that we've wanted from the beginning, from day one, is being able to replace an arbitrary size of DNA precisely at wherever you want, and you can make multiple changes of any type. So you basically use a combination of DNA synthesis, which can be very precise, with insertion, with things like using things that are called recombinases, integrases, and transposinases that have been adapted so that you can program where they go precisely. And so, for example, one of my companies, Poseida, has really exquisite control over where its payload can go. I think that's the future of base editing. It's not base editing anymore. It's gene or genome editing.
Thank you.
Questions? We'll probably take two more.
Hi, my name is Arjan, and I wanted to ask a question regarding xenotransplantation. It's going to go from the more ethical side, as you mentioned, that today we have only like 25,000 transplants per year, and what is going to be the perfect timeline to improve, like, first of all, ethically, how to efficiently produce more pigs for transplantation, and how long it's going to take approximately to make it ideal number per year?
Yeah, excellent question. Our initial set of pigs will be limited by cloning, which is not an impossible production method, but it certainly is currently limiting. We're in the process of switching over to pig breeding, which, as you all know, is very fast and high production. We harvest about, not me, but other people, non-vegans harvest 3 billion a year. They have about 6 months to maturity, about a little under 4 months gestation. But that requires a little more engineering to get to that point. But I think we will be able to satisfy on the order of hundreds of thousands to millions of organs per year. Sometimes more than one per pig. Almost every organ that's currently transplanted from humans can be done with high assurance of no infectious agents and high assurance of it being fresh and healthy, which is not that easy to do with human-to-human transplants. Good ethics question.
George, I have one more question for you from our virtual attendees. Right here, most viral gene delivery modalities are limited by their packaging size. Do you envision a strategy that would allow packaging and delivery of genes greater than 10 kb?
Well, there's already gene therapy delivery, which is on the order of 100 kb, which is the herpes simplex virus, various herpes viruses, all the way up to cytomegalovirus, which is over 200 kilobases. If you want to go even higher than 200 kilobases, I would recommend cell therapies, and we know how to get megabase payloads into cells. And in principle, the entire 6 billion base pairs is subject to change in a cell therapy. So, and this is not that far off. The stuff that, these bigger payloads. Quite frankly, there are not that many strategies that require the big payloads. I mean, the only one really right now that I know of is this pig transplant strategy, which requires genome-wide editing. It doesn't require editing every single base pair of the 6 billion base pairs. Good question.
Perfect. Thank you. I think we'll end the keynote here. Again, George, thank you so much for being here and for speaking with us. Can we just join in with a round of applause?
Well, good luck to you all. I love what you're doing here with openness and maker culture. So take care, everybody.
Thank you, George. Okay, now we'll be moving on to our workshop sessions. We have in-person workshops from Harvard OpenBio, MIT BioMakers, University of Rochester iGEM, and Stanford Space Initiative Biology. So please refer to the workshop sheets in your emails for room assignments. Members of OpenBio and MIT are also outside with signs directing you where to go. For our virtual attendees, you'll find the relevant Zoom links on the sheet that was sent to you. At 11:30, lunch will be catered from Felipe's and Otto's. It'll be served in the cafe area near the atrium where you guys were registering. For our Zoom attendees, please take that time for a lunch break. Return to the same Zoom link at 12:15 p.m. EST. Thanks, guys.
Hi, we're going to get started again. We hope you enjoyed lunch. It's an enormous pleasure to introduce our second keynote speaker at the conference, Dr. Reshma Shetty. As a brief introduction, Dr. Reshma Shetty co-founded Ginkgo Bioworks in 2008. As president, chief operations officer, and member of the board of directors, she's seen the company grow to over 1,200 people. Reshma has been active in the field of synbio for nearly 20 years, co-organizing the first international conference in the field in 2004. So we're following in her footsteps here today. In 2008, Forbes named Reshma one of the 8 people inventing the future. And in 2011, Fast Company named her one of the most 100 creative people in business. In 2014, Ginkgo became the first biotech company to participate in Y Combinator, or YC. In 2018, Business Insider named her one of the most powerful female engineers. In 2019, Bio recognized Reshma with the Rosalind Franklin Award for Leadership in Industrial Biotechnology and Agriculture. And finally, in 2021, Ginkgo Bioworks became a publicly traded company on the NYSE with the stock ticker DNA. Since 2022, Reshma has been a member of the U.S. Department of Defense's Defense Science Board. Reshma has a bachelor's in science in computer science from the University of Utah and a Ph.D. in biological engineering from MIT. Please give a warm welcome to Dr. Reshma Shetty.
So thank you very much to Ayush and the team for inviting me to be here today. It's a real pleasure to join you all. So when they invited me to come, they told me to kind of give you a little bit of a mixed science talk and kind of background on myself and whatnot. So this will probably be a little different from some of the other science talks you've heard today, but I have included a little bit in there. And please, feel free to interrupt me with questions if you have them, during the talk. I'm happy to take them along the way. Okay, so I thought I'd talk a little bit about kind of how I see the field of synthetic biology evolving and kind of a little bit about what we're doing at Ginkgo. I titled my talk, I know this is the Future of Biology conference. For me, my belief is that the future of biology is data, and I'll talk a little bit about that, and it's probably a theme you've heard of earlier today as well. The lawyers make me put this up, so I'll just keep going. Okay. So I probably don't have to convince all of you of this, but the range of applications for biology is expanding. Obviously, lots of people all over the world always think about medicine when they think about biology, but I'm sure most of you understand that the applications of biology go far beyond that, right? The biology is our food, it's our environment. Increasingly, it's what we can build building material, or what building materials can be made from. We can grow our clothing from biology. And increasingly, we have to think about how we're going to actually mitigate climate change using biological solutions. And so this is part of what inspired me when I was a student at MIT to fall in love with the field of synthetic biology, is that there are not so many disciplines who can touch every part of our lives. And so that's why I'm personally, have spent the last 20 years in synbio. Okay, so at Ginkgo, we started Ginkgo when I was a graduate student. There were 5 of us who started the company. 4 of us were graduate students, and then my Ph.D. advisor, Tom Knight. And the first thing we did when we started the company is we came up with our mission statement. So even before we had a name, even before we had a business model, even before we had any idea of what we were going to do, we came up with our mission statement. And at Ginkgo, our mission is to make biology easier to engineer. Okay? And even to this day, we start every town hall at Ginkgo and whatnot with this statement. And so all of our choices along the way, both on the commercial side and on the technical side, are really guided by this particular mission. Okay, so what does Ginkgo do? If you've never heard of us, basically our customers outsource biotechnology R&D to us. And so we work across a lot of different industries. So if I start here on the right side of the slide, we work in agriculture in projects with a lot of the major ag companies in the world. We work in industrial biotech on food programs and sort of shifting chemical production processes to be bio-based. And then we have increasingly more and more in the last few years worked in pharma. So basically helping various companies either in the drug discovery process or in manufacturing optimization, right? And so a lot of folks are stunned when they think about the range of application areas we work in. And the reason it's possible is because we're not a product company. We're not bringing products to market in each of these. What we're doing is providing the innovation engine, right? Folks are outsourcing their biotechnology R&D to us, much like today, technology companies outsource cloud compute to, AWS or Google or Azure. We want companies to be able to accelerate their product development by outsourcing their biotech R&D to us. Okay. Now, I think one of the most exciting things that's happened in synthetic biology in recent years is really the advent of AI. So a lot of you will probably recognize this is Jensen Huang, the CEO of NVIDIA. You maybe have heard of them these days, right? And at a recent talk that Jensen gave, he made this statement. Let's see, do I have a laser pointer? Yes, I do. Okay. Where do I think the next amazing revolution is going to come from? And this is going to be flat out one of the biggest ones ever. There's no question that digital biology is going to be it, right? I think by digital biology, he really means kind of synthetic biology, right? Biology has the opportunity to be engineering, not science. When something becomes engineering, not science, it becomes exponentially improving. It can compound on the benefits of previous years. So this is really awesome to see Jensen say this, right? And because I deeply, deeply believe this as well, that everybody's talking about AI, right? And everybody's going to like ChatGPT and typing in cool prompts and getting cool answers back, and that's awesome. But AI, I think, will have an even bigger impact on biology than it does on human language processing. Now, the challenge with AI is really that we need data to train the models, right? The reason that ChatGPT is so good is because they had many, many rounds they have just a ton of training data. Basically, the entire internet is the training data for ChatGPT, right? And then they did a lot of human reinforcement learning on top of that. And so the question is, can we get equivalent amounts of data for biology so that we can build the ChatGPT equivalent for biological design? Well, this is actually a pretty big challenge, right? Now, the big advantage we have is we can build upon all of the genome sequencing projects that have come before us. At Ginkgo, we've been collecting a lot of proprietary genome data sets as well, mostly candidly through acquisitions. And so we're trying to amass as big of a training set as we can to train either DNA foundation models or protein foundation models. But like ChatGPT didn't just stop with the internet, right? You also need like ways to do reinforcement learning to make these models really, really good, right? And so that means that any company that wants to make use of AI for biology needs to be able to generate data, right? Have access to a data engine. And so long before AI came along, Ginkgo was already thinking about the fact that like really data is the limiting factor when it comes to synthetic biology and biology R&D, right? And if you think about it, we see sort of examples of this all over the place, right? If I just think about, maybe you've heard like several years ago, a group out in California called JCVI, the J. Craig Venter Institute, they built like a minimal cell, right? Roughly like 500 genes or so. And they were basically trying to get it down to what's the minimal number of genes you need for life, right? But even in that minimal cell, which was made as simple as we possibly could make it from an engineering standpoint, they still have like 20% of the genome that's of unclear function. And that problem just continues if you go up in complexity from this very, like the simplest cell we can imagine, like all the way up to humans and beyond, right? Similarly, if you think about the problems that folks in biopharma are facing today, so a lot of interest in cell therapy these days. Some of you might be familiar with chimeric antigen receptors. They're kind of the key like technology that underpins cell therapy. Cars are basically a series of domains, right? And from a design standpoint, what you'd really like to do is be able to individually optimize each one of these domains, but you'd also like to be able to optimize them in combination. That means that's a huge combinatorial design space, and you're trying to pick the one that will work in your cell therapy. And so what you'd love to be able to have is a lot of data on how these different combinatorial designs work so that you can train your AI models to give you better cars, right? And so there's just kind of this general bottleneck around data generation in biology. And so at Ginkgo, we've really been trying to like solve this bottleneck. Okay, so we've been building a set of capabilities for over many years now to just be able to generate large quantities of data. This is just kind of a sampling of those, everything from RNA-seq to proteomics to metabolomics to DNA synthesis, genome resequencing, and then increasingly, more and more work in the kind of biopharma space. So, we need like characterized capsids if you want to be able to do gene therapy, right? And we've been trying to increase the throughput by which we can do experiments just because we need a lot of data to be able to train our AI models. And I really think that AI is a way to unlock possibilities in biological design. And so this has been a real focus for us. We've mostly to date used this internally to be able to deliver on cell programs for our partners. But with the advent of AI, there's more and more interest we're seeing from our customers in being able to access this to generate data to train their AI models. Okay. And so how do we do this? So I'll give you a little bit of a peek and just a few vignettes about how we think about conducting these high throughput experiments and generating the type of data you need for reinforcement learning for AI models. First is we invest really heavily in automation. And I'll talk to you a little bit more about how we do this. Because automation is actually a real challenge. There's a lot of folks who kind of talk a good game about automation, but actually employing it usefully in service of biological experimentation turns out to be much more challenging than you might expect. And so we'll go into that a little bit more. Okay. So in order to do biological experimentation and generate the types of data sets you need for training AI models, you really need to be able to have flexible lab automation. This is almost kind of an oxymoron, right? If you think about how most biological experiments are done, this is what I did during my PhD. It might be what a lot of you are doing if you work in labs, right? Manual pipetting at the bench. I spent about 10 years of my life doing this. I learned a lot, but let's face it, it's kind of painful, right? And it's kind of low throughput. But what it is super flexible, right? I can go in, I'm a smart person, I'm trained at how to do pipetting, I can do a different experiment every day, I can change it on the fly, it's super adaptable to the specific data I want to be able to generate in a given day, a given week. It's just low throughput, right? Now at the other end of the scale, you have what are called integrated work cells. You'll see these at certain pharma companies. Like if you have a lockdown protocol for what you want to do and you want to scale it, integrated work cells are the way to go. You can you can just scale up to tens of thousands or millions of experimental data points generated from this. However, it's probably going to take you 6 months, 12 months, maybe even a couple years, depending on how sophisticated of a workflow you want to do, to be able to fully automate that and get to that scale. Which means that if you then want to make a small tweak to your workflow, good luck. You got to go back and spend another 6 months retweaking your integrated work cell to be able to accommodate the new workflow. And so this is kind of the conundrum of automation, is that either you can have like the flexibility of manual, or you can have integrated work cells, which are really scalable but super locked down. You can do stuff like walk-up automation that's kind of in between, where it's like you're automating parts of it and it helps. But again, just like ends up being a little challenging to really be able to like do complex workflows in a flexible way. And this has been a long-standing challenge, and we've really, candidly, hit up against a wall for a while at Ginkgo and thinking about how to solve this. But there's now a new technology, and this actually came in via acquisition. So this was developed by the folks at Zymergen, and the reason we bought them is really for this technology, because we were so impressed by it. Is what you'd really love to have is flexible, scalable, modular automation. Essentially what the folks did, and we're building on this work now at Ginkgo with the same team, is they thought about, I'd like to break out of this paradigm of flexibility versus scale, and how do I get both, right? And how do I design my automation to support both? The way they did it is they came up with this concept of a reconfigurable automation cart, right? It's literally a cart, right? Inside you can put a single lab instrument. It also has this little arm here that can basically transfer plates from the instrument that's inside the cart onto a magnetic track that can go between these, right? And actually, they even designed them to have little plugs here to be able to hook up individual rack modules to each other, okay? And so the idea is if you have a workflow, you just figure out the set of instruments you need to do that workflow, and you can literally wheel these things up and plug them in together to create like a sequence of a workflow that you want to be able to do, right? And you have you can do HEPA filtering, so you can do mammalian workflows, et cetera, and stuff too. You have utilities going in there, all the practical stuff you need, okay? And then what's interesting is you take this unit and you can start to connect them together, right? And so you can set up an entire work cells here. But because each of these racks is reconfigurable, it's movable, right? You can actually swap these around if you need to, right? Do I have it in here? And so you can even build even more complicated systems of basically integrated work cells of integrated work cells of integrated work cells if you want to be able to scale up either the complexity of the workflows you're doing or the number of different pieces of equipment you need for a particular workflow. Okay, so this has really been exciting for us. Let me go here. As an example of these types of workflows in use at Ginkgo, we did a project with Biogen where we were optimizing AAV titers for them, right? And so we basically onboarded some qPCR workflows onto a rack work cell, and we were able to massively speed up the rate at which we could do that project and achieve the goals of the project for Biogen at the end. Similarly, we're doing similar things with another partner called Microba, where they just want to be able to generate very large data sets, and whether it's high-content imaging, transcriptomic data, metabolomic, excuse me, can't speak, data, and again, leveraging automation to be able to deliver on the scale of data and the diversity of data that they needed. Okay, and so what's cool is that if we compare like a workflow that's done kind of by hand or on kind of more standard like walk-up automation to what can be done on racks, you can actually reduce the amount of hands-on time by kind of 80 to 90%. This is huge, right? If I could take an experiment that would take me all week and reduce it to a few hours, that's a huge savings of time. I can go spend my time actually thinking about the science and not thinking about the pipetting, right? And so this is our dream. Now, all of our workflows are yet automated on racks, but like this is where we'd love to go, right? And what's also interesting is that you can actually build the software in so that you can allow for more parameterization of your protocols. This is the other place where oftentimes integrated work cells tend to fail, right? If you want to make a tweak to your experiment, change the conditions or whatnot, you're rewriting your code. It's a bunch of work to retest everything, make sure it works. With this, they've developed a software to be able to do, first of all, dynamic scheduling, so you can actually interleave workflows on the same racks, right? Which is pretty cool. Just being able to use the work cell more efficiently, and you can also basically like tweak the parameters as you go if you want to be able to adapt to new workflows. So this is, I apologize, I'm like nerding out a little bit on the automation, but like for folks who want to be able to generate data at scale, which I think is increasingly what's going to be needed in order to train AI models, like these are the types of problems you need to solve to be able to generate data at scale. Okay. And so what's cool is that we're seeing this show up. If we just look at, in aggregate, all of the campaigns we run at Ginkgo, when we say campaign, we mean a design, build, test, learn loop at Ginkgo. We run a lot of these, right, across all of our portfolio of projects. And we can look at, essentially just take the expenses of the company and divide it by the number of campaigns we ran in 2022 versus 2023. We were able to reduce our average all-in cost per campaign by about 50% year over year, right? And so we don't think that the end is in sight. This is that compounding advantage that Jensen Huang was talking about in his quote. Like if we can keep investing in the software and automation to be able to generate biological data at scale, you can basically ask more and more complex biological questions. Okay. So let me give you a few vignettes of how we're leveraging this type of technology to actually kind of solve real biological problems. The first one I'll talk about is leveraging AI to kind of optimize enzyme function. So a lot of our projects come in and either our partner has like say an enzyme they use for biocatalysis and they want to improve its function, or maybe they're interested in making a biochemical, in which case it's a multi-step pathway. We need to improve the functioning of each individual enzyme in the pathway in order to improve flux and therefore hit the target titer, rate, and yield that they need from the bioprocess. Right? So how do we do enzyme optimization at Ginkgo? Well, there's a little bit about how we used to do it and how we do it today, right? So we have access to a lot of DNA synthesis capabilities at Ginkgo. And so typically what we would do is take a synthetic metagenomics approach. So this was pioneered by Chris Voigt's lab many years ago now. But you can basically go and leverage available sequence data, right? And basically synthesize a bunch of variants from either the public sequence databases or, in Ginkgo's case, also from our proprietary sequence databases, and then just synthesize them all, transform them, assay them all. And you get a nice data set. And hopefully the winner works, right? And that's typically how we would do it in the past, right? But now with the advent of AI, we can say, well, let's take that data, and instead of just taking the winner from that screen, let's use the data to actually train an AI model and see if we can get even better versions of that enzyme, right? And so why is that important? Well, protein sequence space is just enormous, right? The estimate is that like protein sequence space is about 10 to the 35th. So even if you bias yourself with nature, like you may or may not be able to find the true winner in that huge space. And so you want to be able to kind of learn what is the relationship between function and sequence to be able to find kind of the peak in that very complex landscape, right? Biological design is essentially a search problem. It's searching through this vast space and trying to find the winner. And so high-throughput experimentation can let you search that, but with AI, we can do an even better job of searching that space intelligently. And so what we do is basically we source orthologs, as I said, from nature. We screen them all. We find a lead enzyme. We then plug all of that data, even from the things that don't work, into our AI models. We can generate another round of variants to test. We actually can do that multiple times. And each time we are feeding that data into the model to be able to make that, AI model even better, right? Okay, and so we've done this a lot, across a lot of different enzyme campaigns, across a lot of different classes. Just to give you a flavor of how well it works, these are 3 different enzyme classes that we've worked on. The x-axis here is successive campaigns, right? Successive rounds of enzyme optimization we've done. The green line here is the increase in activity of the enzyme, the fold improvement across the rounds of campaigns. The other y-axis is actually library size, so that's the gray line here. And so what's interesting is you start out with a large library size, you get that data, but then in successive rounds, sometimes you can actually decrease your library size because you're making use of the AI models to basically train and be smarter in picking new enzyme designs, right? And so in this example here, we were able to improve the oxidoreductase activity by more than 80-fold, right? And these plots on the side here actually show you the average mutational depth. And so you can see with successive rounds, yeah, you might be decreasing library size or keeping it somewhat, flattish, but you're actually going deeper into space to look for, deeper into that search space to look for improved enzyme activity, right? It used to be that we would just do this first round and hope for a winner, right? And then maybe we'd have to do it a couple times. But with AI models, we're really seeing the benefit of just how much the leveraging AI can drive improved enzyme function with these successive rounds of generating data, training your model, generating a new set of designs, generating data, training your model. These successive rounds become really more important, and you can actually shrink your library sizes in the successive rounds and still get improved activity because the model is more intelligently searching that biological design space. Yeah. In these types of cases, we are measuring the activity of the enzyme. So the question is, are we measuring folding, or are we measuring actual enzyme activity? And these are almost always activity assays. Okay, so that's the first example. Second example is leveraging AI to optimize RNA therapeutic design. Okay. A typical problem that we might be facing at Ginkgo in kind of the RNA space is, okay, I want to improve mRNA stability and or improve protein expression from the mRNAs, right? And so typically, the way we would kind of approach this now, given today's tools, is make a pooled library of designs, right? And then we'd actually take that data from that pooled library and, again, train an AI model to be able to generate yet better designs. So same principles, but applied to a totally different space. Here are mRNA stability rather than enzyme function. And I hope to show you that basically similar approaches work well in this very different problem set space. Okay. So here's an example where we generated a pooled library with different 3' UTRs. Right? And so it was a pooled library, and we did in vitro assay to kind of look at mRNA stability. And so you can see across that pooled library, you get different designs, right? So here's our benchmark. Here are different designs that show improved stability. Great. And once upon a time, we might have just taken the winners and proceeded. But instead, we're taking this large data set from all of these different designs and the different stability data we see at the end of our assay and plugging it into an AI model, right? To learn from this full data set, okay? So then we go back and we basically use the result of that AI model to try to actually get better and better at predicting how different RNA designs are going to behave in our assay, okay? And you can see, this is basically comparing predicted versus measured. We can get better after successive rounds of kind of generating data, training the AI model, and then generating new data. We can actually get better at predicting how different designs are going to work or not in this assay. So the question is, could we actually take those designs and actually see something that works better in vivo, say in a mouse, right? And the answer is yes. So we were able to take a Ginkgo design and compare it to our benchmark design after a mouse experiment here. right? And you can actually see a pretty significant improvement in the stability of the Ginkgo, of the designed mRNA relative to benchmark by using these kind of successive rounds of training your AI models, generating new designs, and improving them over iteratively, right? So again, same principles, but totally different biological design space. As a last, we're also trying to use this in CAR-T design, the problem I spoke about earlier, right? So we've gotten pretty good at generating a lot of CAR designs. So we can generate kind of 10,000 member libraries here. And we've actually previously reported out and shared data on how well those then CAR designs actually work in vitro screens. But if you're in drug discovery, you sort of don't put a lot of weight on in vitro data. And so the question is, could you do something similar and actually screen a large library of designs in vivo to actually get something that's closer to what might be relevant in the clinic? And this was a work we did, not on our own, but in collaboration with folks at the University of Wisconsin here. Okay. And so this is actually, so the folks at Wisconsin basically had a model for a pediatric cancer, a neuroblastoma. And so these are CAR Ts that are designed to hit the GD2 plus antigen. And so essentially what we did is we applied this library to mice, or rather the folks at Wisconsin did, and they looked at recovery of barcodes, okay? And you can see that you get good recovery even up until injection here. And so, this is not a totally trivial experiment to do, and this is actually like kind of an early instantiation of it. So this is really just pilot data that we're showing here. And then we looked after several days, like day 23, at how much could you recover from the mouse from the tumor, right? And here we can see that basically there is a certain set of CAR designs where actually, even though you maintain the library initially, you see this fall off as you'd expect because only certain designs are going to work, right? But some of those you can actually consistently get back from not just one mouse, but 2 mice, 3 mice, 4 mice, or 5 mice, right? Indicating that they are really actually working those designs and repeatably targeting the tumor, right? This is preliminary data. There's a lot more to do to kind of validate this here, but again, it suggests that these pooled approaches, being able to really search deeply into biological design space, can kind of generate the data that hopefully then we can now feed back into kind of our models to generate even better designs in the future. Yeah. It depends on the experiment. So typically with the enzyme function type of stuff that I was showing earlier, it might be plate reader type experiments type of thing. Here, in this, it's a luciferase assay. Sorry, I should have pointed that out when I showed it. I apologize for that. And then here, we're just looking at recovery of barcodes. So it's like an NGS assay. So it depends on what you're trying to measure. I apologize for not being clear on that. So one of the things we're now interested in doing is actually opening up our platform to others, right? So that others can now generate the data they need to train their own AI models. So, historically, a lot of our work has been done in the context of kind of what we call full cell programs. We're kind of doing the end-to-end work for our partners. But increasingly, as we realize that more people want to be able to take their designs and leverage Ginkgo's platform to test them, we're now introducing a new offering that we're calling Lab Data as a Service to be able to train your own AI models. So this is a new offering. We're really excited about it. If by chance any of you, go off and start companies and want to leverage this in the future, or want to even do it from your academic labs, you're welcome. Okay. So I'm really excited about the potential of synthetic biology, and I dream about what if you could grow anything. I hope all of you are as well. And with that, I'm happy to take any questions. Yeah. Any questions?
But just in the medical industry, I feel like you were talking about the whole. Is there a button? Hello?
There you go.
Okay. So like in the medical industry, I feel like it's just very beneficial for like just like hospitals should just throw out genetic testing, so then they can get like a bunch of data for like some kind of AI model, so then they can kind of like start like connecting phenotypes and genotypes together. So do you have like an answer like why aren't hospitals doing that yet? Is it just like a cost issue or something right now?
Yeah, it's a good question. It is outside my field, so I'm I would say don't take my answer as definitive, but I think it's kind of probably a similar like data depth problem, right? Like humans are complex, our genomes are pretty big. Actually finding correlations that are truly like causal it can be challenging, right? Particularly for complex multigenic phenotypes.
Right. Thank you.
Do you want to catch the catch box? Okay.
Yeah, thank you very much for this talk. Like, it's really like eye-opening in a way, right? I just wanted to ask like sort of like about the quality of the data, right? How like, you know, how can we be, you know, sure, right, that the data that like, you know, the methods that we are using, right? So like what approaches do you think will like improve like the, you know, our sureness in this data to actually then go on and do the applications basically?
Yeah, it's a good question. Yeah, that is actually one of the things you need to be really careful about when you're either doing pooled experiments or very high throughput experiments, is that there can be a lot of, like, you have to have a lot of controls, like far more so, I would argue, than you need when you're doing by hand experiments to account for things like plate effects or, you know, systematic biases in how you set up the experiment. So actually being able to design assays in high throughput with all the appropriate controls to look for any systematic biases in how you've set up your experiment is actually really, really important. And that's actually, I didn't talk about it in my talk, but it's actually like one of the kind of core like pieces of know-how that kind of distinguish folks who can do really rigorous high throughput experimentation versus folks who can't.
Thank you.
It's a good point.
Also, if people want to say like their names and like where they're from.
Sorry. My name's Megan. I'm from Northeastern University. I actually had a similar question about the data quality and how Ginkgo goes about what quality control mechanisms you use to determine the quality of data.
Mm-hmm.
And when do you know if you need to throw something out and like not include it in the pre-training data? I know that's more of an ethical question too when it comes to building AA models.
Yeah. So fundamentally, like our final quality control is like, can our customers actually replicate the same results that we got, right? So that's why actually I would argue there's like a bigger emphasis on quality at a place like Ginkgo than that many, because if you screw up your own experiment and that you just redo it, right? But like we can't hand something back to the customer that's flawed. That generally creates a lot of angst, right? And so ultimately our quality control is like, have we documented both what we did and how we did it, and actually have something in the result that replicates in our customers' hands? So I would say that's like the final like authoritative measure of like quality. But in order to ensure that, you really have to be systematic along the way in how you do your experiments. And so, for example, I talked a little bit about plate effects. Like you need to make sure like not all of your controls are in the last column of your plate. You need to sprinkle them randomly, and you'd actually generally even use software to sprinkle them randomly throughout so that you avoid like location-dependent effects, or that if something's getting measured earlier in the experiment versus later in the experiment. Like part of what I showed of like recovery of barcodes, like are you losing out on barcodes just because of something about like how your experiment is set up, or are you able to maintain them until the actual selective step of your experiment? So like details matter here. It's hard for me to like give you like how we would do it in every case, because it really depends on the particulars of the experiment and what you're doing. But like all of these things kind of end up mattering a lot for generating high quality data. Even like when you first onboard a new workflow, like a new experimental workflow onto the racks, like we go through phases of like, just validating that the instruments are all working as we expect, and then rounds of biological validation before we ever get to like actually running an experimental workflow on like a new like rack system, for example. So there's a lot of this, and I won't pretend we're perfect. We made a lot of mistakes along the way, but like this is something we systematically try to get better at over time, because it is really important to being able to get out good data that you can then use to train your models.
Thank you.
Okay. Last question.
We have like, I think there's like 3 questions
wait,
here.
It's so hard to choose. Okay, wait, you're up front.
Thank you for your time. My name is Naila, and as a data scientist myself, I was wondering how are you compromising the AI models and bioengineering in your company, because the way you are introducing AI to your projects is that AI scientists are coming to your office, for example, and making AI models for you through neural networks and other methods, or you're purchasing already trained AI models from different organizations like Google Cloud?
Yeah, so we're kind of doing all of the above. So we have an internal effort to develop our own foundation models. And we actually announced a collaboration with Google like last year so that we could access the compute we need to actually be able to train our own foundation models. We also take other people's models that are available so we can benchmark and compare the performance. And then we also develop what we call task-specific models, where either using somebody else's foundation model or our foundation model, we're actually doing kind of taking the data sets we have to do like reinforcement learning for a particular task. But we're also, as I showed it, like if other companies have their own AI models that they want data for to train, and maybe they've generated those themselves, they can just come to us and use us to generate data. So we kind of do all of the above, actually. And yeah, so we're happy to have folks use our AI models, but if they want to develop their own and just use us to generate data, we can do that. Or if they want to use their own data and Ginkgo generated data and Ginkgo historical data to train their models, like they can also do that. So there's like lots of options.
Thank you.
Yeah.
Okay, yeah, perfect.
Can I borrow the catch box?
Yeah.
Yeah, of course.
Okay, so we have a question from Zoom. This one is from Amal El-Tayeb. He says, "Hi, Dr. Shetty. Earlier you talked about how machine learning can guide enzyme improvement and design. How are enzyme targets developed from racks being used to study oncogenic enzyme and kinase cascades? Also, do you see data from racks intersecting with public data sets in the future?"
So I don't know that we've actually used, I don't know that we've done work in that particular area. So I can't actually, I think the answer is we're not yet. What was the second part of the question?
Do you see data from racks intersecting with public data sets in the future?
Yeah, it's a great question. I didn't include it in this talk, I don't think, but yeah, we've been kind of assembling, like we leverage the public sequence databases a lot. We've also been assembling our own set of proprietary like genome and metagenome data to basically be able to amass more data to train particularly the foundation models. And so, yeah, the part of what we're trying to do is basically make these tools available to anyone who wants to kind of work with us. And so if folks have their own data sets or public data sets that they want to like merge in with Ginkgo data sets to be able to better train models, like we're happy to do those experiments. I think we'll learn a lot from seeing just like what works and what doesn't. Our hypothesis is that more data means better models, but we need to like fully prove that out.
Cool, and we'll take one more virtual question before we turn it back to the in-person audience. Another virtual question from Anubhav. He says, "Is Ginkgo planning on developing or leveraging AI to improve gene delivery to hard-to-reach organs like the kidney?"
Yeah, I think it's kind of an obvious potential use of like, could you use these types of approaches to like address like tissue tropism and stuff for gene therapy. And so, yeah, I think that's very much like on our radar as a use case.
Fantastic. In-person questions?
Thank you so much for the great talk. I have a question regarding your integration of machine learning models to kind of like search into the sequence space for like more activity. And my question is like, do you have a pipeline that is kind of in a general, like use a generalized model that can fit different enzymatic classes? Like if we take like enzyme specificity for an example, as a way to search for, you have a structural data and that could enhance the activity of the enzyme. Do you have something like that, or you have different pipelines for different classes?
So yeah, so we are building protein foundation models, but in each of these cases, we're also then kind of doing essentially reinforcement learning using the data for the specific enzyme class to basically tune the foundation model for that particular like application. And that's kind of what we've seen works with ChatGPT and stuff. You need to get the really high end performance, you need not just to train it like on like the internet, right? You need to, and Wikipedia and things like that. You also need like the extra levels of reinforcement learning to really like optimize the performance. And so my expectation is for the foreseeable future, you'll need both the kind of large kind of data sets to train the foundation models, and then you'll need to do essentially reinforcement learning in order to get like your models to work well for a particular task. Will at some point we have models that are good enough that you don't need the task specific? I don't know. My suspicion is that there's always going to be a new frontier, like a new task that your model is not optimized for, that you then need to go get new task specific data to train. But that's a hypothesis. Like all of this is like needs to be fully like proven out. But that's kind of what the science say.
Hi. That echo. Hi, my name is Marshall. I had a question about the RACs. I was wondering what their capabilities are now, like how complex of tasks can they do now, and what goals do you have for the future when it comes to this automation?
And how are you measuring complexity? You mean like number of steps in a workflow?
Yeah. How many racks can you, I guess it can go into two questions. How many steps can a single rack do? And how many racks can you hook up together?
Yeah, so you can sort of think of a rack as like the unit of a rack is a piece of equipment, okay? So you're gonna keep basically one piece of equipment inside of a rack. And so in terms of like the number of times you use that piece of equipment in a workflow, it can be however many you want. That's not really a limiter. And so like if you want to think about complexity of racks, you say like, well, how many racks can you put into a system? Like I can't remember what our largest is. I think it's on the order of like tens. So I don't think we haven't yet gone to like 100 rack system, but like we want to. And then you can also ask about how many like different instruments have you onboarded onto racks, and the answer is not yet enough.
Okay, wait. Okay, hi, I'm Vivi. I'm from UC Santa Cruz. And I was wondering, considering the fact that noise a lot of times affects the accuracy and predictability of AI models, and the fact that biological data can sometimes have a lot of noise that can be both human-made and just because of the nature of biological functions.
Yeah.
If providing you, in the case that you get training data from a customer, does Ginkgo go in software-wise to like maybe normalize that data or reduce some of the noise, or do you work with the customers to like maybe produce a different set of training modules?
Yeah, it's a good question. So I'll maybe answer it on a couple different levels. So you're right, like biological data is noisy, and these models, although they can narrow in on like parts of biological design space that are promising, like at the end of the day, you have to experimentally validate every design, right? Like you can't get away from that. We are not at the point where our AI models spit out an answer, a single answer, and then it works, and you don't even have to worry about testing it. Like you're actually spitting out a library of answers, and you're testing them all, and you're hoping that some fraction of them work. So just to calibrate on where we are. But that's kind of like where we are with ChatGPT, right? It spits out an answer, and you say, do you like it or not? Like that's what they do when they do like reinforcement learning. So that's okay. that's still good. It's still helpful. In terms of the other part of your question, which is like, okay, if customers come to us with data they have, and then are we like validating it? I would say like we will definitely look at that. Like it's still early on in terms of our, like we haven't done that like with dozens of customers, so I don't really have an answer to your question yet. But what we have seen that is the closest analog in the past is like when we were doing like full cell programs, customers would give us like assays, or they'd give us like starter enzymes or starter strains, and they're like, hey, this assay works this well, or hey, this this enzyme already has our intended function, can you just optimize it? And like are there like plenty of times when we're not able to reproduce that? Yes, like that is a problem. And so I think what you're highlighting, it will in fact be a problem. And that's like part of what we have to kind of learn, and why I think data generation will be so important is because generating data that's of the form and the quality that is actually useful for training AI models, I think is actually a non-trivial task. Like you can't just take any data set and use it to train an AI model. You really have to be intentional in how you're generating the data so that it's useful for training your AI model, in my view. Again, I'm giving you like opinions versus like stated scientific facts or proven scientific facts.
Go for it. Great. So we have a question from Bhavya Krishnan, who's asking, hi, Dr. Shetty. Thank you for your presentation. You mentioned ML and enzyme improvement. She has a sort of specific question and a broad question. The specific question is, has Ginkgo done research into optimizing enzymes for blood-brain barrier permeability? And then more broadly, how do you think machine learning can be used to optimize small molecules? And I guess the real question is that how is that different from optimizing proteins, for example?
It's a good question. So no, we haven't done research around blood-brain barrier permeability. In terms of using AI to optimize small molecule, that is, I assume for like drug discovery purposes, yeah. So that's not a space we work in because we're like a biological design company, and so we focus on improving biological designs. I am sure that like all the big pharma companies are thinking about how to take, if they can, their historical data around small molecule design and use that to train AI models for like, chemical design, but that's not an area that I work in, so I can't give you the latest on where that is.
Cool. And we have another anonymous question. It's, hi, Dr. Shetty. Thanks for the incredible lecture. My question was, do we have an idea of how much more depth of data we require before we start to see these biology AI models, I'm assuming they're talking about foundation scale models, becoming available to the general public or customers like a ChatGPT for biology?
So there are some that are already available. So I think Meta put out models. So just like there are certain there are like, human language AI models that are made available. There are some publicly available like biology like AI models like out there already. So
cool.
Like, for example, for protein structures.
Fantastic. And I'll ask a question myself as sort of a natural follow-up to that. So folks in the AI field, for those of us in the room who are in touch, are really excited now about what they're calling agents, agent systems, multi-agent AI models, i.e. AI models with the ability to do perception, reasoning, interaction, memory, so on and so forth. And some are even saying that this is a step on the road to self-driving labs. If you're working with racks currently, how far away do you think we are from these sort of self-driving lab systems? What do they think they look like to you?
Are you saying essentially a closed loop system where like the AI generates designs, it runs the experiments, and then it generates a new set of designs, and we all just like sit on a beach somewhere?
Yep. How long before we go out of business, Reshma?
It's a good question. There's a saying when it comes to scientific predictions that people tend to always underestimate or overestimate what can happen in the next 5 years and underestimate what can happen in the next like 10 years. I would say I would go with that. It's like maybe it won't happen in the next 5 years, but I think I don't think it's out of the question by any means.
With that optimistic note, I think we'll close out the keynote. Please give Dr. Reshma Shetty a big round of applause. So we're going to take a roughly 5-minute break. Please, if you can, hang around, stay in this room. Our next panel, featuring Dr. Ritu Raman, Dr. Andrew Berry, Dr. Omar AbuGoot, and Dr. Jonathan Gutenberg, will get started in 5 minutes. Thanks so much.
Okay. Hi, everyone. Thank you so much for coming back. I think we're going to get started. So welcome to the panel portion of our conference. My name is Audrey. I'm a junior at Harvard College. I study molecular and cellular biology and government. Today's panel is going to focus on emerging frontiers and interdisciplinary synergies in advanced therapeutic technologies, which is quite a mouthful. I'm very glad I just got through that. And I have the pleasure of introducing today's panelists, Dr. Gutenberg, Dr. AbuGoot, Dr. Berry, and Professor Raman, which is not in order, but we will start with Professor Gutenberg. So Jonathan Gutenberg is the co-PI of the AbuGoot Gutenberg Lab, a member of the faculty at Harvard Medical School, and an investigator at Beth Israel Deaconess Medical Center and the Center for Vaccine and Virology Research, with affiliations with the Department of Stem Cell and Regenerative Biology at Harvard University and the Department of Genetics at Brigham and Women's Hospital. His lab explores new approaches that leverage programmability across biological scales, including new nucleic acid medicines, molecular tools, and machine learning methods, and applies them to the development of new therapeutics for genetic disease, cancer, and aging. Dr. Gutenberg has co-founded Sherlock Biosciences, Tome Biosciences, and Acquired Proof Diagnostics. So can we please give a welcome to Dr. Gutenberg? We also will be introducing Professor Omar AbuGoot as an assistant professor at Harvard Medical School, investigator at Brigham and Women's Hospital and Mass General Brigham's Gene and Cell Therapy Institute, and faculty member with the Department of Stem Cell and Regenerative Biology at Harvard University. He also directs, unsurprisingly, the AbuGoot Gutenberg Lab, which is developing next-generation gene editing, gene delivery, and synthetic biology technologies using protein engineering and artificial intelligence, and applies them towards new therapeutics and the study of aging. And he has also worked on co-founding Sherlock Biosciences, Tome Biosciences, and Acquiring Proof Diagnostics. So thanks for joining us, Dr. AbuGoot. Next, we have, with an undergraduate degree in zoology from Oxford and a Ph.D. in evolutionary genetics from Princeton, Dr. Andrew Berry originally came to Cambridge, Mass. As a Harvard Junior Fellow. He is currently assistant head tutor of integrative biology and lecturer on organismic and evolutionary biology here at Harvard. His expertise is in population genetics, where he is interested in applying genetic and statistical approaches to detecting adaptive evolution, instances of positive natural selection in genomes. He is the author or co-author of several books, including DNA with James Watson, celebrity, part history, part exploration of the controversies swirling around DNA-based technologies. This was written to mark the 50th anniversary of the discovery by Watson and Crick of the double helix structure of the DNA molecule. Welcome, Dr. Berry. And last but not least, we have Professor Ritu Raman, who is the Diabolov Career Development Assistant Professor of Mechanical Engineering at MIT. Her lab is centered on engineering adaptive living materials for applications in medicine and machines. Professor Raman has received several recognitions for scientific innovation, including being named to the Forbes 30 Under 30 and MIT Technology Review 35 Innovators Under 35 lists, and is the author of the MIT Press book Biofabrication. She's passionate about increasing diversity in STEM and has championed many initiatives to empower women in science. Thanks for joining us, Professor Raman. Okay, so we're going to pose a few questions to each of our panelists, and then we'll be turning it over to some audience questions at the end. I think I would like for our first one to just be kind of going down the line, hearing from everyone what they think the future of biology might look like. And if you could sum it up in a word and kind of elaborate on what you think that means, that would be amazing.
I have to. Well, yeah. It seems completely inappropriate that I say this is the future of biology. I'm the past of biology. No, one word: exciting. That's why you're all here, right? This is a field which has grown exponentially, partly courtesy technological advances, partly due to demographic changes, partly due to epidemics, pandemics, right? I mean, that's changed the landscape. We're at an amazing place in biology, and these guys are going to talk about it to some extent. Some of the technology, some of the methodology, some of the future is blinding. It's a fantastically exciting place to be.
All right. One word is tough, but I think I'll go with optimistic. I really like sci-fi. I hope a lot of you guys do, but I always talk about the fact that whenever we see stories about like the future of biology or robotics or all of these things, they're like kind of dystopian stories, right? Like something goes horribly wrong and then like we didn't think about all these things, but I think what's really nice is that we're in a place where a lot of people are thinking about the future of biology, also have really beautiful, optimistic, like positive views of how it's going to shape the world and how to use it carefully. So that's how I feel.
A word probably would be accessible. I think that you see the bar of what you have to meet to actually make a biological product or drug lowering somewhat. And when I was an undergrad, we certainly didn't have like conferences for undergrads, like OpenBio conference. So the concept of how can we reduce the cost of capital, so to speak, or that level of that anyone could make a biological product, just like people can make a tech company out of their garage. I mean, there's very few undergrads who drop out to form bio companies, and that's not a coincidence. I mean, it's hard to do biology, but that's not a law of biology that it's hard. And I think that new technologies and new approaches and new structures can help us make it easier to make a drug. Not maybe as easy as making a fintech SaaS company, but it can still be, I think, a lot easier. And so it'll be more accessible to everybody to be able to kind of pursue all these exciting and optimistic areas.
Yeah, I would say I'm also, I think, excited about the future. I would say we're really early, I think, in the story of biology. I like to say, I feel like, related to silicon and like computing technology, I feel like we're like in the 50s, where we're just starting to learn how to control cells and proteins and how to, design them using advanced tools. And, we still have a long way to go, right? There's thousands of genetic diseases, thousands of common disease, and even after you cure all disease, right, how do you augment human function? I think we're starting to see the beginnings of that, like these weight loss drugs, for example, that are taking the world by storm. Like, it's kind of crazy. You can suppress appetite and make someone lose, 25%, 30% of their body weight. How do you take that kind of innovation and do that for like cognition, memory, preventing dementia, muscle, et cetera? And even going a step beyond that, right, like with AI, right, like there's this whole idea of how can you augment the human brain to keep up with advanced, thinking abilities, right? So I think there's a lot to do.
Thank you all so much. Dr. Gutenberg, I wanted to come back to what you said and hopefully give you a chance to talk a little bit about, in your opinion, what some of the most promising advancements in molecular bio are that can lead to improvements in disease treatment and diagnostics in the near future, and how do you see that tying in with accessibility, either from the perspective of like scientific opportunity, but also accessibility of care and treatment for all those who are seeking it?
Yeah, that's a very simple question. I think that there's a lot of kind of emerging things that make it easier, hopefully easier to develop, especially therapeutics and understand biology. I do think that when 20 years ago, when the Human Genome Project was like really kind of starting to wrap up, or just wrapped up, there was this concept that it would basically unlock all of these questions about who we are and what the disease is. And I think that obviously not all diseases are cured, and they weren't cured 20 years ago, and they aren't cured now. But we are starting to kind of understand better how these large data sets and how these large compendiums of things can actually help us cure disease. And the Human Genome Project for today, this is obviously a source of debate, but one interesting aspect is that now we're cataloging not just genomes, but the diversity of cells within organisms, especially humans. And this is interesting because this data is quite useful to actually be able to train models. And I think that being able to say biology is advancing kind of as a silo, we want to, I mean, AI and ML are kind of buzzwords these days, but it is true that if you look at the impact that AlphaFold has had on protein structure prediction, there's been a lot of hype, but it is materially good. So how can we use other data sets to understand biology? And I think being able to model these, what happens to biology when you would put a drug on a cell, for example, that's something that I think, in the next decade, we'll have incredible inroads to be able to do. And what that means is that we can start to understand, okay, if this therapy, I want to be able to make this cell go from, a cancerous cell to a non-cancerous cell, or an aged cell to a young cell. Now you don't need, hundreds of thousands or millions of dollars of capital equipment and a screening platform. You could do some of that, not all of it, but some of that in silico. And this also accelerates drug development. This allows us to have new inroads for therapeutics. It allows us to say on the diagnostic level, potentially, what does expression of certain genes mean with respect to disease? So I think the better aspects of modeling will be important for both canonical development of therapeutics, especially, but also making it easier for people who don't have access to a multi-million-dollar lab to be able to run questions and simulations, which you already see with AlphaFold. I think the other aspect of accessibility and molecular tools is that things like DNA, right? If you wanted to make a cool project, let's go away from therapeutics, but there's these glowing plants you can buy now, which are really cool. Yeah, me too. Yeah, no, they're neat. It's but if you wanted to do that, right, you'd have to have your lab where you can clone and your botany area and all this stuff. Why can't we just get DNA in the mail like we can with a printed circuit board, right? You used to have a fab to make a PCB, and now you can actually go and get those online, and it's pretty easy. So I think that as a lot of these services become more centralized, you can't obviously go to a website and buy DNA. It'll just cost you thousands of dollars. As that cost goes down and it becomes easier to do these designs without having to necessarily do all the work in between, it'll make it easier for people at a much lower budget to be able to pursue these ideas and once again make it more accessible for everybody.
Yeah, that sounds great. Thank you so much. And you mentioned a lot of things about like the new technologies and the new types of machinery that are at the forefront of biology right now, which I think is a lot of what's driving the field forward. And I think that kind of ties into something that I want to ask Dr. AbuGoot, which is when you work with things like genome editing and molecular biology, how are you balancing the ethical considerations that are arising with a lot of that? And as a researcher and a scientist, what kind of guiding principles do you follow in this regard?
Yeah, I think that's a great question. This is something that was of a lot of concern, I guess, in the 2014 to 2016 timeframe when a lot of these programmable genome editing tools were being developed, and it was actually making it quite easy for anyone in the world, to edit a genome or, edit a virus. Like, there's a big concern now after the pandemic, like, could anyone just make a new, pathogen virus that could be 10 times worse, right, than SARS-CoV-2? So I think, in our own lab, like in an academic setting, there's a lot of rules and regulations that you can follow to, stay abreast of this stuff. And of course, like, when you're ordering DNA, there's actually a lot of checks that go on behind the scenes to make sure you're not ordering stuff that could be dangerous and whatnot. I think the challenge with AI that we're seeing now is you're actually able to even more easily design stuff that could be, problematic, that could get around safety checks that, people have in place. And so there's even now a renewed interest in how do you keep track of this stuff? Because, while a lot of this stuff can be used for good, obviously any technology can also be used for bad. Ethically, I think, we're mostly focused on, medicines and improving the human condition. So I think, that's kind of our guiding light. And I think as long as you do it in a way that's safe and follows the rules, I think, you can be on this morally correct side, right? So, to give an example, I think there's a lot of interest in installing different types of mutations, right? That can you see in the population, there are mutations that give people lower cholesterol, there are mutations that give people better metabolism and cardiovascular health. And I think one of the challenges, you could easily be like, well, let's install this in everyone. But one thing you need to also consider is when a mutation like that is naturally occurring, it's in the background of someone's entire genetics. And so a lot of times variants in one person might actually be detrimental in another person. And that's like, even when you're thinking about doing good, some of the stuff you have to think about as well. So being really cognizant about this stuff, I think, is really important.
Yeah, thank you. Totally true. There's like so much complexity in biology and the way that a lot of science manifests when you're moving from the lab bench to something that's like a medical treatment or something in a human body. And so this kind of throws it over to Dr. Raman, and I'm wondering if you can describe in greater detail how the living materials that you work with in your lab, first of all, explain what they are, and then kind of talk about how they've developed and changed over time.
Yeah, okay. So maybe I'll level set for the audience a little bit first. So I'm a professor of mechanical engineering at MIT, right? So I should tell you the very little biology in my background. The last biology class I took was in high school, AP Bio, junior year. So that's the level of accessible knowledge that is very useful. So the kinds of things that we're working on in my lab are thinking about how engineers can use cells to make machines, materials, systems in the same way that we use wood, metal, plastic, ceramics, that sort of thing, right? So we are trying to think about how to use that. And so the question was like, how have we thought or how have those sort of living materials changed over time? And my lab is fairly new, but the sort of main thing we do work on right now is we think about, well, what are the things that robots need? They need to be able to sense, process, and respond to their environment in real time. So we focus on like, how do we make really efficient muscle actuators for robots so that they can walk around and move and jump and do things, but use many of the nice advantages of living materials, like the ability to exercise and get stronger or heal after damage. So over time, how this has changed, right, is that I started off basically being like an engineer who the only biology I knew about was muscle biology, and that's the only thing I tried to learn about. As the things that are doing have started to get more robust and useful, we've started to think about, okay, well, how do we integrate neural control in these muscle actuators so that we can have robots that sense a variety of different types of stimuli and make some sort of decision. So you can imagine a robot that's looking around and then decides I'm going to go here versus go there. We've also started leveraging a lot of genetic engineering tools to do things like make our muscle tissues optogenetic so that we can control them with light rather than some sort of very complex, bulky electronic situation. We can also do things that could change protein expression in the muscle to enhance their performance. So you change their fatigue behavior and endurance response. So all of these things are kind of how the living materials are changing over time. I basically say we started off with one cell, now we're going to multicellular complex hierarchies and using tools that these folks and others are developing to really kind of make exactly what we need.
Cool, thank you so much. And throwing it over to Dr. Berry, who I think comes to us with quite a different perspective. This is more from like the education side of the future of biology, and pretty much all of us on the organizing team of this conference have had the deep privilege of being your student at one point or another. And so,
you mean dragged through one of my courses?
Well,
yes,
you said it, not me. But in all your years of teaching at Harvard, in the UK, and Istanbul, what do you think makes a good student of science? And you're here with a lot of phenomenal scientists. And so what do you think is the difference between a good student and a good scientist? And are all good students of science meant to be good scientists? Are there like particular attributes that you expect to be seen in the next generation of leaders? And what do you think is going to push us forward in the future of biology?
Well, the first observation is there isn't, you might expect there to be good concordance between good student, I've done well in all these courses, I understand all this science, and a good scientist. And there's an overlap. That Venn diagram stippled area certainly exists, but it's not in any sense direct concordance. That's because there is more to being a scientist than getting an A in a course. And I've known plenty of students, yes, in that got an A in the course sense of student, it's gone off into the lab and it's not gone well. You need, it doesn't mean to say they necessarily burned their hands off with acid. I'm just saying it didn't work out. So what are the attributes that take, yes, it's a sort of prereq. You've got to know and love science, but what's the extra? What's the extra ingredient? Breadth. I think one of the things you don't want to be is just too narrowly focused on your thing, because you're not going to bring anything to it. All you're going to do is be drilling down ever deeper into wherever it is. We just heard a lovely story about starting off as a mechanical engineer, needed to learn something about biology, so learn something about biology. So there's a perfectly good model here of research knowledge which is as needed. But what you need is the breadth of background to know that it's needed, to know that it's out there, to make those interdisciplinary connections. So I think breadth is critical. And the other thing, this is a personal thing for me, I think a respect for nature. And I'm not going to go off on, I'm not going to start talking about endangered species, don't worry. By that, I mean, think about several of the major advances in the past 20 years or so. Obviously, CRISPR is a biggie. Where did that come from? It didn't come out of the brain of a very smart person. It was basic biological research exploring the natural world, right? We stumbled across a bacterial immune system. So that's part of the breadth. The breadth is interdisciplinary, but it's also a recognition of the fact, yes, I'm a neuroscientist, but there's lots of really interesting biology out there, and I need to fold that into my research experience, if you like. That, plus, of course, a whole bunch of rather boring personality traits like willingness to take risk, being an innovator, having a vision, all those things help, of course. But no, interdisciplinary breadth and a respect for what nature has achieved in a mere 3.9 billion years. That's what you need.
Thank you. That's profound. So I think interdiscipline has clearly been a large theme of this conference, and it's very clear that a lot of the prominent advances in biology are not just rooted in biology or in the classroom, but in connecting with a lot of other things. And so, Dr. Raman, since you've had a chance to talk about some of your research, I'm wondering if you can talk more specifically about biofabrication in future biological and biomedical innovation, especially in neuromuscular tissues and beyond, and maybe talk about one of your favorite projects or advances that is currently going on.
Yeah, I mean, I think that sometimes, we do things or talk about things in a certain way, not because that's the only thing we're excited about, but also you want to convince people that this is something meaningful to do, right? So that they give you money so that you can do it, right? So sometimes when we talk about the work we're doing, we say, hey, I'm very interested. My fundamental reason, right, is that I think the way that humans and other animals and everything in our natural world navigates very unpredictable environments. I think the motor control system that we use to do that is very exciting, and I want to learn more about it, and that's my reason. But then I need to give an additional reason, usually, to convince people to do the kind of work, right? So I say, hey, when anything goes wrong with that system, that really sucks for you, right? Because you have a limitation of your mobility, which really impacts your health and quality of life. So if you want to give somebody sort of the most agency often over their natural and built environment, you want to make sure that they can navigate their motor control system. So often kind of early ways of how we were describing our work or thinking about the intersection of these fields was to say, we want to make engineered neuromuscular tissues so we can use them as platforms to understand more about diseases that restrict mobility and use them as cell-based or tissue-based therapies. And one of the most exciting studies that kind of came out of that most recently from our group was showing that by using the optogenetic tissues that we developed, we could implant muscle grafts in a mouse, exercise them transcutaneously through light stimulation, and this not only helped the grafts sort of mature faster and get stronger, but also increased the rate at which the host animal was able to grow new vasculature and new nerves into the tissue. And that was very exciting and was kind of in medicine, but then kind of thinking again about being at the intersection or interplay of these fields, we've now been able to use that learning that exercising muscle can be used to enhance nerve growth to be able to integrate neural controls into the muscle actuators that we deploy in robotics. So there's always this back and forth of fundamentally, you have a question you're interested in. Depending on who's funding the research or who you're talking to, you can say, oh, it's really cool for medicine, or it's really cool for robotics. But every time you learn one thing in one domain, something fundamental about biology and the way like intercellular crosstalk is governed by exercise, you can use that as a tool or technology to push forward the more like engineering applications of the work. And there are other times, of course, that things that we've learned on the engineering side of things have fed back into biology. So it's really nice, and I would like to sort of double click on what Dr. Berry said about not specializing too early. I think it's super, super important to have a breadth of knowledge before you dive deep into anything.
I think it's super interesting that you can stand at where you are in biology and kind of look back and see how everything that you've done in the past has been building up to this. And I think Dr. AbuGoot got to talk a little bit about the development of genome editing. And I'm wondering if you can cover a little bit on how you think the field is going to evolve in the next decade. So maybe ideas on what kind of novel like molecular tools are going to come into play, and maybe a more extreme version of ethical concerns, or like what do you think the big ethical questions of the next 10 years are going to be alongside the new tools that are inevitably going to be developed?
Yeah, I think that's a great question. I mean, for one, I think CRISPR, got a lot of attention and hype over the past decade, but I'm not sure if people fully appreciate this, right? There's, 7,000 genetic diseases, and, we just got kind of the first approval like 3 months ago for the first one, right? Like sickle cell, right? So there's actually a lot of work to do to actually apply the technology now to the rest of these diseases. And it's a tough challenge because, for one of the technologies that really needs to be developed is delivery technologies, because the sickle cell thing that just got approved is actually not in like a drug you inject in someone. They take stem cells out of your body, they edit them with the CRISPR tool to correct the sickle cell mutation, and they put those hematopoietic stem cells back in the person, right? So this vision of being able to inject something in someone's body, have it go to the exact cell types you want, correct those mutations, and treat someone is still not fully realized. And there are some companies that are working on that, right? Like Intellia has actually an in vivo genome editing drug for this amyloidosis disease. And the reason they're able to do that is because that's a liver disease, and most things you inject in the body, at least with gene editing reagents with lipid nanoparticles, end up in the liver. So then there's a question of, okay, well, what about all the diseases in the brain, the heart, spleen, kidney, lung, et cetera? And so, there's a lot of work that needs to be done to develop, whether it's lipid nanoparticles or viruses that can hone to those tissues and deliver these reagents. So I think that's one big thing. I think the other thing that needs to be really developed is probably innovations in manufacturing or other aspects of developing these technologies, because it's really expensive to actually develop a drug, right? Like the sickle cell drug, I forget what it's exactly priced at, $2 million a person? Let's say it's $2 million a person. Most of these gene editing drugs end up being $2 million, $3 million a person, and that's because hundreds of millions of dollars actually go into funding the companies and development of these things, because it's really hard to do these. And so, there has to be innovations in making, these things easier. Some of that has to do with the cost of DNA, the cost of synthesizing RNA, the cost of doing mouse and, primate studies. But I think until that happens, it's going to be hard to imagine bringing this to all the diseases. And even once you get past 7,000 genetic diseases, there's then, how can we do stuff? I mentioned this at the beginning, but how can you implement genetic edits that maybe make you live longer, right? Or that can treat obesity, cholesterol, heart attacks, and atherosclerosis, right? And so now you're thinking about scaling this up for even more people. And so additional innovations, I think, are needed there as well. Yeah.
Thank you so much. And I'm really glad that you brought up aging, because I think that transitions really well into my next question for Dr. Gutenberg, which is, in studying aging from a more scientific perspective, what do you think are the most promising molecular tools that you've developed for understanding cellular dysfunction and selectively eliminating detrimental cells? And how do you see these tools impacting regenerative medicine, either just in the moment or also in the foreseeable future, the next 10 years?
Yeah, so aging is a complex, not disease, phenomenon is perhaps a better way to put it. And I think that if you compare aging to cancer, right, we know, we can define, I think, a pretty good consensus of what cancer is at different scales, right? You have a dysregulation of cell growth that leads to formation of tumors, and those invade and lead to patient death. Aging is not so well defined because it's a much more complex system. And so this makes it of course, difficult to develop therapies. So the role of new technologies is twofold. One, as we learn more about aging and age-associated disease, how do we treat those? But of course, how do we understand what aging is? How do we understand the dysfunction that occurs during aging? How do we measure those things? How do we perturb them? So I think one thing that we're very actively exploring in the lab is that if you take young and old tissues or cells from a diverse number of patients, how can you understand what is changing at the molecular level? Can we sequence, millions of cells from different patients at different ages? And that gives us, clocks. You may have heard of this methylation clock, if you're familiar with it. It's a way to measure how different marks on DNA change over time. It's a little phenomenological. We understand what RNA does, so if you look at transcription and regulation, that's actually, I think, a much better way to understand it, so making those sorts of clocks. So that's one start, but then the second thing is, once you have a metric, you can perturb things and see how that metric changes. So a really powerful thing about these molecular tools is that we can do pooled genomic screening. We can change, turn up, turn down, turn off, every gene in the genome across the population. And then we can measure how it changes those clocks, measure how it changes the entire cell transcriptome. So we can do those sorts of measurements and look at which genes are involved in aging, and we're involved in aging in different tissues. How do they change where you are on that clock? So this is a critical thing to understand really what happens across different tissues. And then the cool thing is, we can use CRISPR as an intervention too. So if we see, okay, turn this gene down and it makes HSCs wind back the clock by 30 years to be a little reductive, can we develop that and deliver that, to go back to Omar's kind of point, into patients and actually use that to rejuvenate their cells? And so it's kind of nice to have the ability to manipulate in the lab and the ability to manipulate in the clinic have a similar modality. So I think that it's critical to be able to use these to understand. And then the last thing I'll say about aging is that obviously aging is very complex, and what as evolutionary defined aging is, there's a lot of questions about what are the evolutionary limits on aging, et cetera. But I think one thing is that by using these tools, we can overcome biology in a lot of ways. If there's a gene that is important in development, then, and that leads to loss of years at the end of your life, obviously that'd be selected for, but we can edit it, later on in your life. Or if there's a gene that's important in some cells, but not in other cells, we can manipulate it using a lot of these programmable tools, delivery, other things, only in certain cell populations. So we don't need to follow the rules of, the genome is the same in a majority of your cells, just counting your immune cells. We can edit in particular contexts and use programmability to actually bypass a lot of those traditionally hard set rules by evolution that can lead to new therapies. So I think that's why we're very excited about how we can apply this to much more complex situations like aging and age-associated disease.
Yeah, the idea of bypassing biology is, I think, really incredible, and I love that. It's very hard to separate what happens in research and in the clinic. And yeah, I really just love the way that I think we've been framing all of our innovations in the context of the human body and what that looks like in the future. And you bring up methylation, and I remember learning about methylation in a certain somebody's course. To all you Harvard freshmen out there, good luck on your final. But this one goes back to Dr. Berry, and I think that as an academic, but also as very clearly a mentor to the next generation of scientists building the future of biology, what do you prioritize in your teaching, and what do you think is important for all of the young scientists in the audience to learn in their journey to becoming revolutionaries?
Well, the answer to the first part of the question is simple. What do I prioritize in my teaching? Is a desperate attempt to keep students awake. That's my primary priority. The idea is, and it returns to what I was saying earlier, don't rush into narrowness too fast. So the course you're referring to is a course in genetics, genomics, and evolution. That's an awful lot. So it really is an incredibly superficial skip across a vast amount of science. I think that's great. I think that's super appropriate at the undergraduate level because you get the overview. And you might think, okay, we understand genetics and genomics come together. Why are we doing the evolution thing? Because it turns out the profile of genetic variation in the human population, of course, is a function of the evolutionary demographic and population genetic history of humans. In other words, how, where, and when we evolved. So that's the thing, that's the beauty of this, of an interdisciplinary perspective, is everything does actually touch on everything else. And we really should run a long way from siloing things into separate categories. But I'm just going to make another general comment about overcoming biology. I'm going to push back on overcoming biology. So the joy of biology, let's talk about aging. I mean, we die at whatever, 82 or whatever is our current mean age for expiring, and we can drag that out for a little bit longer, yes. The Greenland shark, I think, has a mean lifespan of 465 years. There's enormous variation in nature in the aging process, and there are various theories as to why some species last longer than others and so on. But we can't just learn from other pieces of biology, we can take from other bits of biology. So that's the joy of comparative biology, is we have an opportunity to see what a genome that doesn't age at the same rate as us, what it does, how it maintains itself, and so on. And I'm saying, so don't go, yes, we want to push beyond our biology, but let's use biology to push beyond our biology.
I think it's a great point. I mean, on that, like, not only like necessarily learning from, like, you know, long-lived species, but there was a study where they took, like, naked mole rats, they live like 30 years, right? Much longer than like mice, right?
You've got to tell them not everyone knows what a sexy little beast a naked mole rat is.
You can Google image search it, maybe. I mean, they look fine. They're just like a little like hairless. There was a study where they took a certain protein, I think it was like a hyaluronidase or some structural protein, from those into mice and actually made them live longer. So we don't necessarily have to learn from the sequence and like the actual biology. We can literally take, right, genes from other organisms and put them, not that necessarily this is going to be a therapy for humans in a while, but we can, I think that's a great way to also, you know, borrow or maybe steal, depending on which word you want to use, from, you know, across the, you know, tree of life. Yeah.
Awesome. Thank you both so much. And I think that Dr. Berry's comments will lead nicely into my last question for all of you before we turn it over to the audience, which is, what advice do you have for the students here that are pursuing a variety of scientific careers? Anybody who would like to start?
I'll start. I always say the same thing, so that way it's easy for me. You know, I don't have to think of anything new. My advice is always, I think particularly in America, and I think particularly for people like my age and younger, so you guys are much younger, we've been told often to like pursue our passions, right? Find the thing that we're like really excited about and like motivated by, and that's how to like choose or define a career. And I certainly think it's like good to be interested in what you do, but I think that's like a little bit limiting, and often when we're very young, you haven't even seen all the sort of things and you don't know what you could get excited about in the future. So I personally have found it more rewarding to be like, here is a problem that like really affects me, really affects a lot of people, and what skill sets do I have, or how can I use, or how can I address that problem, right? It could be in human health, it could be in climate change, it could be in something else. And that way you're like motivated by a thing that really matters to you and your community, and then the path you take to get there can change over time. So yeah, I would say focus maybe less on passion and more on problem solving and caring about your community.
You can do, I think, two, which is, and these are two from experience that I did not do. So I think you can like learn from that. First is, you know, take risks, but especially don't be afraid of failing. I think even failing, you know, in a public way, right? Going off and building something is a good learning experience. You want to maximize on learning, not necessarily accomplishments. Especially when you're young, failing can be as good as, if not better, than success. Learn from your successes too. But I think the biggest kind of error is to, you know, stay where it's safe and, you know, not step outside of your comfort zone because, oh, this will look bad. You know, in the future I will screw up. I think that's you know, I know many people, myself, you know, self included, in science is like this, where you have to fail to succeed. So yeah, try to fail. And the second thing is related to that, but especially don't set up barriers. I think the most effective people in anything, science or anything, you know, just run through walls. You don't set up barriers for yourself especially, and don't let, kind of other things set up barriers. In grad school, I mean, this was a positive thing. I, went to one institution. I was a grad student at Harvard, but I grad my MIT lab with Omar, and a really great advisor, Fung. But I think the biggest barriers are those we set for ourselves, telling ourselves, I can't do it, or this isn't possible. Figure out a way to do stuff and do stuff quickly and fail quickly, and then you'll be much more successful and be able to iterate much quicker in the long term.
And this sort of pertains to that same point, which is, and I talk to a lot of students who are anxious about setting out on a research journey because what do I know and how useful will I be, and I'm not trained in this and I don't know how to write that kind of code and so on. It doesn't matter. So doing research is, from your perspective, in fact for everyone's perspective, is a learning process, okay? What you're bringing to the table, and research isn't something you forge ahead and do by yourself. It's a collaborative exercise. You're bringing a particular skill set to the table. Somebody else is bringing a different skill set, which you don't have. Somebody else is bringing a third skill set, okay? So that's the important, it is, yeah, it's the risk-taking, moving out of your comfort zone. Don't walk around with a chip on your shoulder thinking, oh dear, I'm not good enough. You're great, actually. The fact you're motivated and you want to do it is literally half the battle. So go for it, is what I'm saying.
I guess my points will be related to the last 2 points as well. I think my first advice is pick hard problems. I feel like it's really easy these days to get caught up in building like the next, AI app for like organizing your calendar or like a social network. Like pick harder problems. I think, in biology especially, there's a lot of really challenging, impactful things to go after, right? Like aging is a great example, right? Like, I think 15 years ago, aging was like a fringe science in biology, right? Like, how, what is aging? How could you study that? But now it's like a really exciting field that's opening up and people are actually making progress, right? And I think there's other areas, right? Like climate change, I think is another really interesting area to apply biology for. Really hard because, how the heck, biology is already hard to engineer. How can you do that now at a global scale? But I think, those are the types of problems that, you guys who are the bright future, right, should be pursuing. And I think that connects to the next point, which is related to all this failure, like don't give up on the hard problems, right? I think, it takes a long time to see progress on a day-by-day basis. It's going to feel like you're constantly failing. But I think, there's always something new to try. There's always new perspectives, new ideas. Sometimes you hit a block and a week later you'll see through it. And so I think if you're going to make, like, everyone goes through this, right? If you're going to make progress on those hard problems, you basically just have to never give up, right? And that's actually the solution to success. So, yeah.
Awesome. That's super promising, and thank you guys so much. I think it's the time of the panel where we'll start to open it up to audience questions. So any of you in the audience, please raise your hand. Feel free to pose it to the panel or a specific member. We will also have a microphone. We have some attendees on Zoom, so speaking into the microphone just ensures that everyone can hear you. So please raise your hand if you'd like to ask a question, and Carl will get it to you. Oh, Carl's taking on the catch box. This is, if you guys don't know what this is, you're going to see why I'm scared of it.
Oh, come on, Carl.
Good arm.
Thank you so much for the great panel. My name is Khlib. I'm a sophomore from Ukraine at Brown University, and I really would like to follow up on the comments that Dr. Berry made about the respecting nature, and because it's really resonates with me. I'm very interested in the natural history of ants, the different species, diversification, evolution, ecology of ants, and I feel that in my field, it's really hard to get funding in general because everything has to be connected to humans, applications to the alive, how it's useful for humans, and because I'm only a sophomore right now in undergrad, it puts some sort of a pressure on me and thinking about, oh my God, in grad school, do I have to change, betray my aspirations, or do I should look at it from different perspective and think, oh yeah, I can research my ants and maybe it will be beneficial for the humanities, stuff like that, which I'm not super interested about, to be honest. I'm just interested in fundamental research. But yeah, I just like to hear maybe your comment on this, sort of the general question is, should we support fundamental research, right? Or should everybody do the fundamental research as a part of their useful research?
One interesting question. Did you say ants? I just want to clear that. I think that's fantastic. I did not expect to meet, and the term of art, ladies and gentlemen, is myrmecologist here. So I don't think you need to compromise. Yes, I agree, funding's hard, but funding's hard for everyone. My attitude about going to graduate school is you go to graduate school to do something that excites you, the very first word I used in this panel, and that'll prepare you for something. Don't go in there with this grand plan, you map out your entire future, I'm going to apply for funding here, I'm going to get this job here, and no, because life happens and all sorts of serendipitous things occur. So I would hope that you could find a path that doesn't involve you sacrificing your love for the tiny creatures, as E.O. Wilson said, that run the world. And again, it's going back to what I said about love of nature. There's lots of amazing insights into human biology. I know everyone thinks, bloody ants? No, that's not a model system. But there's all sorts of interesting stuff buried in ant genomes. So yeah, stick with the ants.
I'll maybe just tack on to that. Don't forget that we all live in a society, right? So it's all of our responsibility not only to do science, but to talk to our friends and family about what we're learning, what was exciting. And that way, if everyone knows why you are excited about basic science, then hopefully eventually we can convince people to fund the sort of things that are there, right? So we have to communicate outwards also.
Okay, hi, my name is Yudong, a freshman from Brandeis University. And this question is mostly directed for Dr. Raman. I'd like to ask, how does your agitators change the way that we approach rehabilitation for trauma or disease patients, such as patients with arthritis? And do you ever see your lab producing any alternative treatments instead of chemotherapy drugs for arthritis patients?
Great, yes, I always start, okay, so first thing, it's ramen like the noodles or the spectroscopy, depending on how you feel like identifying. But actually here, so we should say spectroscopy, we're all scientists. Great question, yeah, so I mean, I think right now, a lot of the things we focus on are making healthy muscle tissue replacements for things that are caused by traumatic injury, so large volumes of muscle or nerves that are lost. Eventually, we want to think about developing grafts and therapies for diseases that have other types of causes, right? Where the muscle itself is diseased, it could be through dystrophies, for which a lot of gene editing and ASO type therapies have been very useful. And it could also be for things like neurodegenerative disease, where people think that often degeneration starts at the periphery and then you have dying back of nerves upstream of the muscle. So that's kind of the level we're thinking. I've never thought about arthritis, but certainly some of our work in the exercise space more broadly has taught us that exercise really, has these huge systemic impacts on the body and really mediates how muscle communicates. Right now, we're focused on nerves because we're a small lab and we have, sort of limited funding and scope. But as we we have huge data sets that have shown that exercising muscle regulates crosstalk with vasculature, crosstalk with the immune system, and crosstalk with other sorts of things that could mediate progression of other diseases. So I think using sort of peripheral tissue to affect things upstream and restore mobility across a broad spectrum of diseases is something we're very excited about.
Just while we're waiting, can I return to the ants? Sorry. This is Omar said it's difficult to think of biological applications, though this is an important problem with climate change, right? I'm going to just give you a very sexy example, very recent. Ants and butterflies, they have, as you probably know, structural elements in their scales which create different colors and so on, and they interact, obviously, with incident radiation very differently. Turns out if you've got desert-dwelling ants, let's say, there's some really subtle things they do with the fine structure of their exterior to keep themselves cool. You can take that idea, this is biomimicry. Thank you, ants. Sorry, we're not giving you any cash on this royalty deal. And you can design a paint that has elements of those same structural elements, which will then, you can paint, and they've done experiments, painting it on flat-roofed houses in Bangladesh in high summer, super hot. It reduces any bills related to cooling by, like, I'm making this up, but 60% or something crazy. That's ant-inspired science for climate change.
Thank you. Other questions?
I have a question for Dr. Abudea. You mentioned for your advice for students to try and solve hard problems. How do you define a hard problem, and how do you try and find a hard problem that's within the scope of your abilities?
Yeah, that's a great question, because I think I actually think like education does not teach how to find problems well, right? Like, you know, I went to grad school actually at both Harvard and MIT, and you know, I did this unique postdoctoral position called the McGovern Fellowship that basically let me run my own lab after grad school. And when I made that transition, I realized, like, you know, what the hell am I actually going to study, right? There's obviously like the extension of my grad school, which is obvious, right? You know, all the follow-up stuff, but like, how do you define new directions? How do you find what's worth studying? How do you find what you can get funding for and what you can convince people is interesting, right? So that process, you know, like they don't teach that, even though I think they should, right? And there was actually an interesting article by, I think it was a Harvard professor. What was that guy? You know what I'm talking about? In Cell?
No. He's at UCSF.
Oh, no, he's at UCSF. That's right. What's his name, though?
He does, not Chris Huttenhauer, but he's the Michael Fischbach.
Michael Fischbach.
Stanford,
Well, yes,
sorry.
that's it. Look up. He published an article recently in Cell, actually, that was exactly about this. Like, how do you find problems? How do you find what's interesting? And I'll just tell you from experience and trial and error over the past 5-6 years, a lot of it has just been, you know, brainstorms we have with our lab, with each other, with other colleagues, right? Ranking, you know, problems by, you know, how many people will it impact? Why will it impact them? Like, how will it impact them, right? Like, you know, aging and climate change. Why did I say those were impactful problems before? Well, climate change literally affects every single person on this planet, right? And we're careening towards potentially a disaster. For aging, also similar, right? Like, how many medical diseases can you think of that actually touch everybody? Like, I guess you could argue, you know, we've all been affected maybe by cancer or something, but I mean, like, actually physically touches everybody. There's not a lot, right? Maybe cardiovascular disease, maybe aging. And so, you know, when you think about problems on that scale, right, that's, you know, partially where impact can derive from, right? So, and then to answer the second part of your question, like, what intersects with your skills? I mean, I would say, like, don't limit yourself, right? Like, you know, Elon Musk, for example, right? Like, was he a rocket scientist? Was he a you know, specialist in battery development, right? He really just thinks about interesting problems and figures out, how do I pool the resources and right expertise to go solve those? And I would argue, even though I did my PhD in bioengineering, the thing I learned the most about that is actually how to be organized, how to do the process of science, how to think about problem solving. And I can apply that, honestly, to any problem inside biology or outside of biology. So, you know, I think there's a lot of problems outside of biology that, you know, maybe one day, you know, I will also go after with the right amount of time and dedication. So, yeah.
Thank you. I think we have time for just one more.
So other than lowering costs, what other specific actions do you think could be taken to make biology more accessible for all? So particularly other than the undergrads in really prestigious colleges and more members of the community?
Yeah, I mean, I think that it's not just lowering costs, but like lowering, you know, what you need access to, right? So not everybody has access to certain equipment or certain labs, and so the ability to circumvent that, right? Where you don't need, you know, here's a key example, right? Right now, if you wanted to clone, you know, a piece of DNA, right? You have to get access to an incubator to, you know, grow that up. You'd have to have an area where you could have bacteria to, you know, put that DNA into. You'd probably need a PCR machine, you know, something that can change temperature. And there are somewhat cheaper, you know, ways to do these things. But instead, you know, what if there are ways that you could circumvent that entirely? So I think that's one thing. Obviously, going back to biosafety risks, those need to be managed as well. But I think not needing these things at all, you know, irrespective of expense is one aspect. And then I think the other thing is that in terms of knowledge, not many people necessarily know how to do these sorts of things. How do you design it, to stitch this together? I mean, I think that it'll become more and more accessible where you don't even need to know the like nitty gritty of these different aspects, just like we don't need to know like TCP/IP protocols to use the internet, right? Or machine level code to write a software application. So I think more layers of abstraction as well. I mean, I'm glad that I had to learn it and it was fun, but it's a barrier, right? And so better abstraction and better ways of modeling these things, when we're designing biology, for one example, will make it easier and easier for people to just go out there and say, I have this grand vision of how I'm gonna reprogram this bacteria to eat plastic, and just do it. I think that going down to breaking down barriers and stopping people from doing what they really want to will be going a long way.
Thank you so much. Can we get a huge round of applause for all of our panelists? Thank you all for an amazing panel and some great discussion. I'm going to pass it off to Hugh to explain our final conference segment.
All right. Hi, everyone. Can you hear me okay with the green box mic? Okay, so this panel has been the last kind of super sit in the lecture program that we have today. The last about hour and a half of the conference is just going to be a poster and networking session out in the cafe where we had lunch and where we did registration this morning. So we have about 10 people who brought posters, and they're very excited to share their work, and you can go and look at that basically in person, talk to them. I think hopefully they'll be manning their posters. We have a number of posters online via Zoom that you can either access based on links that we will send out now, or for those of you who are online, you can access those online most easily. And then there are going to be a few companies and one university who's here to do a little bit of networking if you're interested in learning a little bit more about their opportunities to get involved. And it's just kind of a final way to meet your attendees at the conference and get a little bit of food and really round it up in a good way. So, and just before that, I think I can speak for the entire team, and I know Ayush is going to talk for a second. We're so happy with how this has all gone, and we hope you've had a wonderful time. It's really been a pleasure to see you. Yeah, thank you.
So before we head out to the networking session, just a couple of quick words on behalf of our team at the Harvard Undergraduate OpenBio Lab. On behalf of our collaborators, the MIT BioMakers, first, thank you to all our really esteemed panelists for joining us today. We have a small little tokens of our appreciation for you.
It's not small.
Thank you so much.
Sure, thank you.
And we're grateful to our panelists. We're also grateful to you, the attendees, both in person and all the many attendees we have on Zoom. Like I said at the start of the conference, a couple years ago, we launched the Harvard Undergrad OpenBio Lab with a really clear vision. We saw a gap in support and resources for motivated undergraduate biology researchers who wanted to work both within and outside of faculty-affiliated labs to change the world with biology. This is our second annual Future of Biology Conference, the first time we're able to do this in person, bring you all here together in the Harvard Science Center, and we just couldn't be more thrilled with how many of you showed up, how many of you dedicated your day to us and to our amazing speakers to learn about what the future of biology might look like in 5, 10, 15 years. We are really excited to continue advancing this mission, to grow our fellowships program, to put on this conference every year, to hopefully have our speakers return again and again. And if you are also excited about biology just as we are, please don't hesitate to reach out. Our website is pretty simple. It's www.harvardopenbio.org. You can reach us at exec@harvardopenbio.org. We'd love to hear your feedback, what you thought of the conference, how you think we can improve, and opportunities to engage over the next year. We host, in addition to this conference, pretty much every month, we host workshops with both in-person here at Harvard and often virtual options. We've had really esteemed speakers from places like Nucleate, Strand Therapeutics, Ginkgo, and Asimov come in and speak to students and get them inspired about the future of biology. So the purpose of OpenBio at the end of the day is to provide a platform for inspiration and a platform for action, to take your ideas about the world that you want to see using biology in 5, 10, 15 years and make that happen. We're really grateful also to our partners for the event today. We really like to thank our sponsors, the MCB department at Harvard, the 1517 Fund. We're really lucky to have Danielle Strockman here from 1517 Fund, so thank you, Danielle, for being here. And we are so excited to continue to engage with you. So again, please enjoy the networking session. We hope you had a wonderful conference. And if you have any questions, comments, or feedback at all, please reach out. Thanks for coming.